Indonesia, the biggest archipelagic country on Earth. Made up of thousands of volcanic islands, best known for beaches, volcanoes, Komodo dragons, orangutans sheltering elephants, and tigers. The mega biodiverse country is inhabited by 20,000 different kinds of herbs and plants. Also a place where you can find more than 10,000 kinds of animals roaming the land. Indonesia is homeland to hundreds of ethnic groups speaking as many as 650 different languages. Differences is our motto, made us strong by one vision of the country. Formerly parts of University of Indonesia, in 1963, IB University was stood as the first independent campus to focus on agriculture. Located in Bogor, West Java, IB University brings strong vision as a leading international university focusing on agriculture, marine, and biosciences. There are 11 faculties and schools, comprises 39 undergraduate study programs, 65 master study programs, and 42 doctoral study programs. There are 27 internationally accredited study programs in IB University as a proof of our commitment as a world-class university. IB University, through its performance, able to craft achievements and acknowledged as the most innovative state university in Indonesia. In promoting students' quality outside the class, student club and societies are places for students of the same interests and hobbies to be and share.
Welcome to IB University. We are very pleased to welcome all of you to the 20th International Student Summit in Bogor.
Indonesia, the biggest archipelagic country on Earth. Made up of thousands of volcanic islands, best known for beaches, volcanoes, Komodo dragons, orangutans sheltering elephants, and tigers. The mega biodiverse country is inhabited by 20,000 different kinds of herbs and plants. Also a place where you can find more than 10,000 kinds of animals roaming the land. Indonesia is homeland to hundreds of ethnic groups speaking as many as 650 different languages. Differences is our motto, made us strong by one vision of the country. Formerly parts of University of Indonesia, in 1963, IPB University was stood as the first independent campus to focus on agriculture. Located in Bogor, West Java, IPB University brings strong vision as a leading international university focusing on agriculture, marine, and biosciences. There are 11 faculties and schools, comprises 39 undergraduate study programs, 65 master study programs, and 42 doctoral study programs. There are 27 internationally accredited study programs in IPB University as a proof of our commitment as a world-class university. IPB University, through its performance, able to craft achievements and acknowledged as the most innovative state university in Indonesia. In promoting students' quality outside the cross, student club and societies are places for students of the same interests and hobbies to be and share.
Welcome to IPB University. We are very pleased to welcome all of you to the 20th International Student Summit in Bogor.
Indonesia, the biggest archipelago country on Earth. Made up of thousands of volcanic islands, best known for beaches, volcanoes, Komodo dragons, orangutans, sheltering elephants, and tigers. The mega biodiverse country is inhabited by 20,000 different kinds of herbs and plants. A place where you can find more than 10,000 kinds of animals roaming the land. Indonesia is homeland to hundreds of ethnic groups speaking as many as 650 different languages. Difference is our motto, made us strong by one vision of the country.
Kirain dia udah jadi.
Formerly parts of University of Indonesia, in 1963, IPB University was stood as the first independent campus to focus on agriculture.
Lihat aja nggak apa-apa.
Located in Bogor, West Java, IPB University brings strong vision as a leading international university focusing on agriculture, marine, and biosciences.
Ibis dari pagi ya bu. Pantas pakai ini gitu.
There are 11 faculties and schools, comprises 39 undergraduate study programs, 65 master study programs, and 42 doctoral study programs. There are 27 internationally accredited study programs in IB University as a proof of our commitment as a world-class university. IB University, through its performance, able to craft achievements and acknowledged as the most innovative state university in Indonesia. In promoting students' quality outside the class, student club and societies are places for students of the same interests and hobbies to live and share.
Welcome to IBB University. We are very pleased to welcome all of you to the 20th International Student Summit in Bogor.
North Sumatra, a sign of God's love given with full affection, a piece of heaven attached to the earth, home of the world's largest ancient volcanic lake that holds 1,000 legends and mysteries, a land for diverse cultural conformity where civilizations live in harmony, a place of dignified tolerance where the seeds of hope are planted, grown, and reaped for prosperity. The genuine miniature of Indonesia.
By declaring the tagline transformation towards the ultimate, Universitas Sumatera Utara is ready to transform into an international standards university with excellent local characteristics. I am Murianto Amin, presenting you Universitas Sumatera Utara.
Starting from Universitas Sumatera Utara Foundation, which collected moral and material support from the people of North Sumatra and Aceh, on August 20th, 1952, the Faculty of Medicine was successfully established. USU was inaugurated as the 7th state university in Indonesia by the first president of the Republic of Indonesia, Sukarno, on November 20th, 1957. Located in Medan City, North Sumatra, USU consists of 2 campuses located in Jalan Dr. Mansur and Kuala Bekalah. USU has 15 faculties, 1 graduate school with 163 study programs, and 1 experimental garden covering an area of 300 hectares located in the Tambunan area, Langkat Regency. USU has the vision of becoming a university that has academic excellence as a barometer of scientific advancement that can contend in the global world. Carrying a new spirit, transformation towards the ultimate supports the entire USU academic community to make significant changes to their mindset and work culture per the demands of bureaucratic reform and technological acceleration. Transformation towards the ultimate is indicated by a series of transformations in various sectors. The transformation is realized through collaboration and synergy between the academic community and various parties to create excellent higher education services implemented through Tree Dharma. Campus digitization is an absolute must to provide the best service. Various digital services are provided by USU, such as ASA, e-Office, e-Agenda, PPID, and also telemedicine. Telemedicine provides easy access for medical treatment to the public through their gadgets. USU has received an A accreditation and is included in Cluster 1 of the Indonesian Ministry of Education and Culture 2020 rankings and ranked 15th university in Indonesia by the Times Higher Education Impact Rankings. USU has also established itself as a part of the international education community. USU is ranked first nationally and ranked 305th in the world in the field of agriculture and biological science at the university level by the Schemato Institutions rankings. In addition, USU is ranked 1201 plus in the world, with QS Stars 3 on the Kwok Bareli Simmons World University Rankings 2022 rating agency. As a commercial university, USU is committed to encouraging research to create an USU enterprise by establishing the Institute of Innovation and Business Technology Incubation, or LINCS. LINCS is a place for innovation and research products of USU's academic community to make contributions to society. Some of these innovative products are being prepared for mass production by cooperating with the industrial world and other universities as the implementation of the Merdeka Belajar Campus Merdeka program. Merdeka Belajar Campus Merdeka program is a manifestation of learning by combining theoretical and practical aspects to create excellent human resources. USU collaborates with various partners, from the state-owned company to the government sector, with the aim of creating graduates who are ready and able to follow the needs of the industry and the market. To support comprehensive learning, USU provides integrated facilities. Various facilities available include USU Hospital, Dental and Oral Hospital, Integrated Laboratory, Student Workshop, Auditorium, Student Center, Pancasila Building, International Guest House, Cafeteria, Student Dormitory, Garden, and various sports facilities.
The spirit of transformation towards the ultimate within the scope of the Tridharma is a manifestation of USU's commitment to produce excellent human resources for the advance of Indonesia.
USU believes that the contribution of all parties is the main key in the effort to transform to be superior. USU, the pride of North Sumatra for Indonesia, transforming to be the best in the world.
Indonesia, the biggest archipelagic country on Earth. Made up of thousands of volcanic islands, best known for beaches, volcanoes, Komodo dragons, orangutans sheltering elephants, and tigers. The mega biodiverse country is inhabited by 20,000 different kinds of herbs and plants. Also a place where you can find more than 10,000 kinds of animals roaming the land. Indonesia is homeland to hundreds of ethnic groups speaking as many as 650 different languages. Differences is our motto, made us strong by one vision of a country. Formerly part of University of Indonesia, in 1963, IPB University was stood as the first independent campus to focus on agriculture. Located in Bogor, West Java, IPB University brings strong vision as a leading international university focusing on agriculture, marine, and biosciences. 11 faculties and schools, comprises 39 undergraduate study programs, 65 master study programs, and 42 doctoral study programs. There are 27 internationally accredited study programs in IPB University as a proof of our commitment as a world-class university. IPB University, through its performance, able to craft achievements and acknowledged as the most innovative state university in Indonesia. In promoting students' quality outside the class, student club and societies are places for students of the same interest and hobbies to meet and share.
Welcome to IPB University. We are very pleased to welcome all of you to the 20th International Student Summit in Bogor.
Indonesia, the biggest archipelagic country on Earth. Made up of thousands of volcanic islands, best known for beaches, volcanoes, Komodo dragons, orangutans, sheltering elephants, and tigers. The mega biodiverse country is inhabited by 20,000 different kinds of herbs and plants. Also a place where you can find more than 10,000 kinds of animals roaming the land. Indonesia is homeland to hundreds of ethnic groups speaking as many as 650 different languages. Different sex is our motto, made us strong by one vision of a country. Formerly parts of University of Indonesia, in 1963, IPB University was stood as the first independent campus to focus on agriculture. Located in Bogor, West Java, IPB University brings strong vision as a leading international university focusing on agriculture, marine, and biosciences. There are 11 faculties and schools, comprises 39 undergraduate study programs, 65 master study programs, and 42 doctoral study programs. There are 27 internationally accredited study programs in IPB University as a proof of our commitment as a world-class university. IPB University, through its performance, able to craft achievements and acknowledged as the most innovative state university in Indonesia. In promoting students' quality outside the class, student club and societies are places for students of the same interest and hobbies to meet and share.
welcome to IDB University. We are very, very pleased to welcome all of you to the 20th International Student Summit in Bogor.
Okay, can we start now? Okay, everyone has joined this Zoom. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome back to our program, summer course program. Let me introduce myself first. I'm Muhammad Arifin, a lecturer from IPB University. This summer course is a collaboration between IPB University and University of North Sumatra. I hope you enjoy with this program, and I hope the material history provided by the lecturer was useful for you all. Before we start today's class, I remind to all participants to turn on your camera and then turn off your microphone during the presentation. And then if you have any question or comment, please use the raise hand feature and for to talk directly to lecturer, or write down your questions or your comment at the chat room. Okay. For today's lecture, we will be delivered by Bu Prof. Irma. Bu Prof. Irma, thank you so much for your time. It is an honor for me because she is my supervisor when I studied at master program at IPB University a few years ago. Hello, Prof. Irma.
Hi, good afternoon, everyone. Can you hear me?
Okay, thank you, Prof. Thank you for your attendance and your contribution for our summer course. I want to inform you that this summer course consists of several countries, namely Malaysia, Pakistan, Bangladesh, Ethiopia, South Korea, and Indonesia, of course. And consists of level degrees such as a bachelor, master program, and doctoral program. And a few several is come from general public. First of all, let me introduce yourself, Prof. The committee, can you share the CV, please? Okay. Okay. Professor Dr. Irma Isnapia Arif, SPTM, is Master of Science, is a lecturer from Animal Product Technology, Department of Animal Production and Technology, Faculty of Animal Science, IPB University. Next, please. Yeah, Prof. Irma is a graduate from IPB University for undergraduate until doctoral program. But some her research is carried out of Osaka University in Japan. Is that true, Prof? And then Prof. Irma is very expert in animal product technology field, especially in microbiology and meat science. And then currently, she is a Vice Dean of Animal Science Faculty, and then previously he served as a head of Department Animal Science and Animal Production and Technology. And then please, she have a lot of publication. This is a publication in last 10 years ago. She have a national publication is 53 papers, and international is 60 papers, and her index Scopus is 9. It's a very good thing, and the Google Scholar is 12. And she also have 3 patents. It is very nice. The first patent is the method for production of plantaricin EEA1L5 bacteriocin. And then the second one is Roselle probiotic yogurt formula for health function and manufacturing process. And then the last is the method of making probiotic fermented sauces. If you want to know more detail about that, please you can contact to Prof. Irma by email irfnavia@apps.ipb.ac.id. Okay. Here's a short introduction from Prof. Irma. For the next, maybe I deliver to Prof. Irma. Prof. Irma, time is yours, please.
Thank you, Mas Arifin. Good afternoon, everyone. Can you hear me clearly?
Yeah, it's good.
Okay, I'm so sorry because I am at a hotel now. After continuing a meeting in academic process in IPB. So I would like to thank you to the committee, Summer Course Committee of Department of Animal Production and Technology, who permit me to join this summer course, and then I will share about my projects, my research result, also my competencies in animal product processing, especially in fermentation product, fermented product. So can I share now?
Yes, please, Prof.
Okay. So for this course, I would like to be in dialogue with the students, also with the participants. So the class will be live and also will be more interesting because many interaction between us. So I would like to share about the animal products processing, especially in diversification and functional properties. As Mas Arifin mentioned before, I am from Division of Animal Product Technology. My name is Irma Isnavia. So the outline, will I share now the two parts. The first about the fermented food, also bacteriocin. What is the bacteriocin? I will explain after we discuss about the fermented foods. The second one is non-fermented foods, but only a little part of non-fermented foods. So maybe in the task from the committee, maybe can share about or will give you a task about the specific traditional animal products in your country or in your origin hometown, maybe, yeah. I don't know the task. I will give you a time for discussion after I finish in one part in one topic, and then we can discuss more detail about it. So the first topic is about the fermented foods and bacteriocin. What is the fermented foods? Also, what the beneficial function of the fermented foods? The second one is about the okay. The second one is a non-fermented foods from animal products. Okay, there is Indonesia picture of the food industry. The high market value for the food industry in Indonesia due to the market size. We can okay. You can see in the picture that Indonesia has bigger consumers than Thailand, also US and Mexico. So Indonesia is a high market value for food industry. So don't afraid if we can produce the healthy food, more food for the people, so Indonesia will be not in recession. Yeah? The second one, the picture, we can see the mega diverse countries of the world. What is the mega diverse country? Is the country who has many diversification plant, diversification of animal, also diversification microorganism that has been collected by the researchers and then published in many journals or papers. So we can see that Indonesia is one country, also Malaysia. There is many papers who explore the many traditional food, many traditional local food from many regions in this country. So, for example, in Indonesia, this is in Solo or Yogyakarta town in the central Java. So you can see many foods here. Maybe it is from tempe, fermented soybean, also maybe from the meat, also chicken that has been fermented for many hours, maybe, or very well cooked. So this is many function for our head also. So if we can isolate, we can get many microorganisms from each food. Maybe this is a satay, satay and tempeh. So we can isolate many microorganisms in, for example, bacteria, also yeast, yeah. So Indonesia has many microorganisms that can be isolated. The next slide, we can see animal-based traditional fermented food in Indonesia. Also, maybe in the August, many participants from IPB also visit USU, Medan. So you can see maybe traditional food from Medan. And near Medan, we can find dadia. Dadia is from West Sumatra. It is fermented milk that incubated into bamboo by naturally fermentation, and then we can get the fermented milk, or maybe a soft cheese. I think, yeah. The other one is dango. Dango is a fresh cheese. We can say that from Sulawesi. It's also traditional food, only from fresh milk, and then we can add papaya or some protease enzyme from papaya, and then they can coagulate the casein in the milk, and then we can get fresh cheese. We can say that, yeah. So the other one, for of the fermented meats, we can find in the Bali. Bali has a traditional sausage. It's called urutan sausage, but it's made from pork, yeah. But we can change the materials from the beef, also lamb, maybe chicken also. We can produce the fermented chicken sausage. So it's a unique or traditional fermented food in Indonesia. And I think in Malaysia, also Thailand, we can get more fermented foods originally from Malaysia and originally from the specific country. So the next page, we can move to the basically theory. What is the fermentation and what microbes that contribute to the fermentation process? What is the fermentation? General definition is an aerobic cellular process, but I can also say sometimes aerobic, sometimes anaerobic. If we produce cafe from maybe from rice, maybe we can say that as the fermentation is aerobic, not anaerobic fully. An aerobic or aerobic cellular process in which bacteria, yeast, or other microorganisms convert organic foods into simpler compounds. What is the meanings? From the lactose in the milk to the sugar, to the simple sugar. From the casein into peptide, from the whole of protein into peptide with the low molecular weight. This is the meaning of convert organic food into simple compounds, and chemical energy is produced. And the specific definition in food processing, the fermentation is the conversion of carbohydrates into alcohol and also carbon dioxide. This is the heterofermentative fermentation by organic acid, for example lactic acid, by yeast, bacteria, or combination of both. So the fermentation is very old technological process in food processing, because the fermentation process is inexpensive process. We can add the bacteria, or the fermentation will be continued by natural bacteria that has been in this food. For example, if we will ferment milk without bacteria, it can be happen. But the quality, also the nutrition, also the safety of the product will be damaged. So we can add the specific bacteria to maintain the quality. So this is all inexpensive process of processing vegetables is coming back into fashion. For example, like this, the cleaner and safer than canning. The process also produces the helpful bacteria known as probiotics. For the first step of the fermentation stage, root vegetables, cabbage, and fruits are all well suited for the fermentation. Like the picture in the right, fermented foods in the diet. Okay. We can divide fermentation into two types. The first one, alcoholic fermentation, and the second one, lactic acid fermentation. What the differences between alcoholic and lactic acid fermentation? We can say also homofermentative and heterofermentative fermentation. What is the homofermentation? Homo is only one produce, hetero more than one products. So this is the anaerobic metabolism in fermentation. The glucose will be deferred, convert into the pyruvic acid by the glycolysis, and then convert to the lactic acid. This is the right set is lactic acid fermentation. Only the one product, lactic acid, from the glucose. The other one in the lab from glucose and then continue to the glycolysis mechanism into the pyruvic acid. Also with the pyruvate, the carboxylase will be produce carbon dioxide and then acetaldehyde, and then the alcoholic, the hydrogenase, we can get the ethanol, yeah, for the end product. So this is the second one is alcoholic fermentation, or we can call it the heterofermentative fermentation. Okay, what is the function of fermented food products and their uses? The carbon dioxide, we can get the specific bread using better yeast. Yeah, for example, Saccharomyces cerevisiae. So we can get a good texture of the bread using Saccharomyces cerevisiae. The second one, alcohol for the wine making and brewing using yeast. For example, what is this? Yeah, wine making. Yeah. And the third one is lactic acid. Lactic acid bacteria ferment milk into product as yogurt. The other product is sauerkraut, fermented cucumbers, and kimchi from Seoul, yeah, or South Korea. Also, most studied lactic acid fermented vegetables. So, for the student from Malaysia, you can collect, yeah, fermented food which are originally products in your country, also for the another country. So what is the origins of some fermented food? We can see the region from China, Korea, Japan, North Africa, Europe, Middle East, also Egypt, also Indonesia, also from Malaysia. We can get the tea maybe, yeah, and fish sauce and fermented meat, sourdough, beer, cheese, fermented milk, wine, soy sauce, and mushroom before Christmas, yeah. So the years before we are here, yeah, in the world, yeah. So it's very interesting, yeah. Amazing food. Okay, we can discuss about the benefit of fermented foods. Why we eat fermented foods? Should we eat the fermented foods? I answer should. Should maybe better, yeah. Better you can for a week maybe you choose one of the special day. Maybe you can get and you can buy fermented milk, yeah. Maybe yogurt or another products for the better, yeah. Better you have for your stomach, as your GI tract. You can maintain the composition of bacteria in your GI tract. Why we eat fermented food? For the first one, we can get enzyme. For example, increase enzyme. Where we can get the enzyme from the bacteria. Increased enzyme content helps you absorb nutrients, because the many bacteria also has, for example, yeah, protease enzyme, so that can help you to absorb to digest the protein to peptide, reducing the need for vitamin and supplement. The second one, you can get the healthy bacteria. We can call it the probiotic. This is the good bacteria. So in the for the head GI tract, gastrointestinal tract, or our stomach, our intestine, we should maintain the composition between good bacteria and bad bacteria. The percentage of good bacteria should be higher than bad bacteria. The good bacteria have restored balance in the gut and at digestion and immune health. I will explain in the next slide. The third, should we eat fermented food for get safety of the products? Because the lactic acid created during the fermentation process can kill E. coli. E. coli is a pathogenic bacteria that can damage the food, making it safer to consume than raw vegetables or raw food. The food is for preservation. The lacto-fermentation process holds food longer than canning without depleting nutrients. And the last one, we can get more nutrition value. The fermentation process increases the nutritional value by enriching certain nutrients. For example, the whole protein to be the simple peptide, and then lactose to be the simple glucose, and then for example like that. Okay, this is another picture of the fermentation process. This one is homofermentation, homofermentative, and the second one heterofermentative that we can get in the before. Before I explain this. So, what is the good bacteria? The good bacteria, we can call it probiotics. Probiotics can be defined as live microorganism. It should be live microorganism that we consume in a certain number, that it can give health benefit in our stomach. So the probiotic should be live microorganism. What is the genus or familia or the probiotics? We can get from the lactobacillus. We can get also from the lactococcus, propionibacterium, Streptococcus thermophilus, Bifidobacterium, Lactobacillus bulgaricus. For example, in yogurt, we can get Streptococcus thermophilus and Lactobacillus bulgaricus. Another product of fermented milk, maybe Yakult. Yakult is made from specific probiotics that we can say Lactobacillus casei strain sirota. So it's very specific strain. Okay, most of good bacteria belongs to lactic acid bacteria, yet not all of them are probiotics, because the probiotics has requirements. The first one, they should be generally recognized as safe, species as strain identification. So if we isolate lactic acid bacteria from traditional fermented food in your hometown, maybe, so you should identify by molecular identification, so we can get the unique species and the strain of the bacteria. Not only the biochemically technique is not enough to identify this bacteria. So it should molecular identification, minimally 16 sRNA sequencing. The second one, they should be tolerant to acid and bile because our stomach is very acid. So they should pass the in our stomach and then go to our intestine and go well in our intestine. And in the mid of our intestine, they can get bile salt. So the harder system for probiotics, the first acid and the second one is bile in the intestine. The third, they have antagonistic activity against pathogenic bacteria, so they can go well in our intestine and then they can kill the pathogenic bacteria by producing bacteriocin or maybe lactic acid. And the second one, they could adhere in intestinal cell line so that they cannot only flushing out from our intestinal tract. What is the research? What is the future research? Future research, we can more detail in the exploration of legacy bacteria-based probiotic from many indigenous resources are very important. Okay, this is the effect of probiotic bacteria and yeast in our gut. This one, the screen, the screen is probiotic bacteria, also yeast. Sometimes Saccharomyces cerevisiae is called probiotics, sometimes not, sometimes can be. And then the probiotic bacteria can adhere into the intestinal mucus in our intestinal tract, and then they produce defensin. Defensin is one of the immune substance, and defensin can also catch the pathogenic bacteria that may be adhere also in our mucus. The next, probiotic bacteria can give effects in the mucosal immunity by suppress the B cell in this intestinal to produce IgA. IgA, immunoglobulin A, is specific immunoglobulin that only we can get in our intestinal tract. This is very specific immunoglobulin, so we can say this is the immunoglobulin A, the specific immunoglobulin. The another one, because our probiotics can adhere into the intestinal tract, so they can move. Another bacteria can move from adherence in intestinal tract. Maybe this is E. coli or pathogenic bacteria, or will move, by competing between probiotic bacteria and pathogenic bacteria. Okay, this is the mechanism, another mechanism or detailed mechanism, microbial fermentation and effects on the host functional properties. We can imagine the host functional properties is our intestine, So sometimes we can say that commensal bacteria is probiotics, maybe probiotics. So the commensal bacteria can induce gut development into in the this one in our intestine, and then the other one suppress the B cell to produce immunoglobulin A. Also the other one to the goblet cell also will produce mucus, and the other one also dendritic cell to produce more immune substance for our immune system. Why SCFA? What from SCFA come from? This is the fermentation process that acted by the lactic acid bacteria. So the fermentation will produce SCFA, short chain fatty acid. This is the another mechanism. Okay, so many years ago, probiotics we get from traditional foods, maybe from tempeh, miso, from South Korea and Japan, and yogurt, also kefir. The other one, also we can get more the food for probiotics. We can define it prebiotics. So prebiotics is the food for probiotics. Probiotic is bacteria. Prebiotic is carbohydrates. So this is the sources of prebiotics. Okay, what the slide explain about fermentation as an alternative strategy for improving nutritional status. So this is very good food, many vegetables, and then we can ferment it, into the can maybe, and then we can get the fermented vegetables. Also, we can get maybe fermented food, another fermented food. For example, tempeh, fermented soybean, yogurt, and so on. And from our kids, our children, please give them the fermented foods. Okay, this is I will explain some of types of fermented foods isolated we can get originally in Indonesia. From the first one is dadi. Dadi is an Indonesian ethnic fermented meat from West Sumatra. It's made by pouring fresh cow, unheated buffalo milk, fresh unheated buffalo milk into a bamboo tube. Fresh milk inoculate to a bamboo tube kept with banana leaf and allowed to ferment at room temperature. We don't use specific incubator to ferment it for 2 days, maybe 1 day or for 2 days. So the product is solid like this, and stout. High nutrients contents as compared than fresh milk, because the lactose will convert to the simple glucose. Protein will convert to small peptides, and so on. Okay, this is my breakfast today. From animal products. In the left, chicken dim sum. In the right, dadi, originally from Payakumbuh, West Sumatra. In the middle of July, I visited Padang, also Payakumbuh, and my college, my college brought the Dade for me, and then I collect it and then I bring it into the Bogor, and this is for the next research in our lab, in our laboratory, to isolate lactic acid bacteria originally from Dade, maybe from my bachelor students, maybe this is a topic, research topic for my bachelor students. Okay, the next the next slide is Dango. Dango is fresh cheese, In Indonesia, it is fresh soft cheese that is usually made from fresh cow or buffalo by the farmers' household in Enreka, Sulawesi, South Sulawesi province. Dango made by heating a small with a small fire, then add coagulant from papaya. Maybe we can get papaya enzyme from papaya, and then resulting in natural protein, which change the course of buffalo milk become solid due to the separation of protein and water. So the products has higher nutrients content as compared to the fresh milk. The other one is yogurt. The yogurt is specific name for the fermentation of milk by two lactic acid bacteria, Lactobacillus bulgaricus and Streptococcus thermophilus. If we use the other types of bacteria, maybe Lactobacillus acidophilus, we cannot say this is yogurt, but it's fermented milk. Maybe the name is specific, maybe acidophilus milk or plantarum milk. This is not yogurt. Yogurt is only specific name from fermentation by Lactobacillus bulgaricus and Streptococcus thermophilus. The lactose is fermented to lactic acid, and it's the one which cause characteristic card to form. The pH value is 5.5. So Lactobacillus bulgaricus convert lactose to lactic acid, and then proteolytic enzyme from Lactobacillus bulgaricus break down the milk proteins into peptides. This peptide stimulate the growth of Staphylococcus thermophilus, which in turn produce lactic acid that are growth stimulant from bulgaricus. So this is the mutual symbiosis between two bacteria, coupled bacteria. We can say it the very well coupled bacteria. Okay, this is the type of yogurt: set yogurt, stick yogurt, flavor yogurt, Greek yogurt, frozen yogurt, and so on. You can searching in the Google maybe in the what are types of yogurt? Many yogurts, many types of yogurt. The other one is kefir. What is the differences between kefir and yogurt? Kefir is beverage produced by action the lactic acid bacteria, yeast, and acetic acid bacteria on milk, which produce a distinctive fermented milk product with unique properties. It is produced by adding a starch culture called kefir grains. So the kefir grains is the symbiosis bacteria and yeast. The symbiosis bacteria from the lactic acid bacteria, yeast, and the other bacteria, acetic acid bacteria. The kefir grain are mass of several different bacteria and yeast embedded in the complex matrix. So if we isolate bacteria, it's very hard to isolate it. because it's very specific, yeah, and very strong complex matrix, yeah, between protein, carbohydrate, also the combination between the lactic acid bacteria and yeast also acetic bacteria. It's very strong connection between two types of the microbes. Traditionally, prepare kefir contain 1 until 2% alcohol. Yeah, so the mood of feel of kefir is described to be prickly and sparkly. Yeah, because there is carbon dioxide content, also only 2% alcohol. This is the picture of kefir grains. So the production of kefir grain, how we make, how to make kefir, very simple procedure. We pasteurize milk, and then we add the kefir grains into the milk and cover the can, yeah, and then ferment into the room temperature up to 20 hours fermentation process, and the next day we can get kefir grains, and also we can collect again the kefir grains for the production of the kefir tomorrow. So the kefir grain is very specific. They can grow and make a bigger matrix, yeah, after the fermentation, like yeah. Okay, the next product is cheese. Yeah, cheese sometimes fermented milk, sometimes not fermentation. Okay, yeah. What kind of cheese are you making? Select the first milk. The origin of the milk is the first step in determining the flavor and consistency of the cheese. Although most cheese is made from cow's milk, sheep's, and goat's milk also can be used. Okay, how to make the cheese? Yeah, we already learned that milk has two main parts: the water and solid. There are other names for these two parts. And as milk split into two parts, we call it curdling or cleaving. Whey is the correct name for the liquid. So we can get two parts after the enzyme, yeah, coagulate the casein into curd, and this is cheese. The final product is cheese, and the other one, the byproduct, is whey. Whey is for the liquid one. Curdling happens natural as the milk sours, but it will be done intentionally as the first step in making cheese. This is the natural process. Okay, we can also add the enzyme rennin. Rennin is enzyme obtained from the stomach of young calves, added to raw, whole milk in liquid or tablet form. It causes the milk protein casein to coagulate. Yeah, the clabber milk. So we can cut it with a knife. Yeah, like this. Yeah, so we can get the fresh cheese. Okay. So what is the byproduct of cheese production? We can get many volume, yeah, a big volume of liquid whey. The liquid whey is drained from the curd, which are then crumbled into pieces. If you make cheese at home, you might use cheesecloth to drain the curd. Okay, so the final product, salt, muscat, ready for finishing steps. This is the cheese production, yeah, very simple. Okay. How many times I can finish in what?
You have 30 minutes, yeah. Bro.
Okay. So this is more specific research, yeah, in my laboratory in probiotic application on fermented foods. I selected two products, yogurt and fermented sausage. So this is new, not new, yeah. This is our products. And altogether, the research indicated the some function properties of probiotic rosal yogurt has anti-hypertension effect, anti-diabetic effect, and hypocholesterol effect. And we can know we have the patent certification. Yeah? This is the research of the probiotic rosal effect. The first one in 2016, we can find that antimicrobial activity of goat milk yogurt with addition of Lactobacillus acidophilus 2b4 and rosal extract. So this is the journal from UPM. Many students come from UPM. Yeah, from UPM Malaysia also, yeah. And the other one, anti-diabetic potency and characteristic of probiotic goat milk yogurt supplemented with rosal extract during food storage in Tropical Animal Science Journal 2018. This is the research journey, yeah, roadmap, yeah, of the probiotic rosal yogurt. The other one, this is the explanation. Activities of angiotensin converting enzyme, inhibitor enzyme on probiotic yogurt and probiotic yogurt rosal. That means that probiotic yogurt rosal can maintain the hypertension. So it will be very good, yeah, for the hypertension consumer, yeah. This is the data. We use goat milk, yogurt probiotic, compare yogurt probiotic rosal. The angiotensin converting enzyme inhibition only 20% for the yogurt probiotics and increase if the yogurt probiotic add with the rosal extract up to 38%, yeah. So the angiotensin converting enzyme is enzyme that contribute to hypertension. So if the inhibition of this IC increase, so it will be good for our health. Yeah. The other one, this is. Okay, this is the renin angiotensin aldosterone system. Why it can be happen? So the angiotensinogen is produced by our liver. With renin, it will become angiotensin 1, and with angiotensin converting enzyme come from the lungs and kidney will be converted to the angiotensin 2. And then angiotensin 2 will increase the sympathetic enzyme and reabsorption natrium chloride, also arterial vasoconstriction, and then the aldosterone secretion. So it will be high hypertension. If we block the angiotensin converting enzyme here, we block it. So the angiotensin 1 cannot convert to angiotensin 2. And what the substance can block the enzyme? This is the peptide. Peptide from the fermentation process. The casein will be converted to the specific peptide. But it's come from the fermentation, yeah. Only three amino acid, yeah, combined with the unique sequence. Only this sequence can block this one. Yeah, this is the mechanism. Okay, the next one. Okay, this is the proof that the fermentation process can degrade casein to the low molecular peptide. Yeah, this is the first one. The band, yeah, with the blue, yeah, the blue one is this casein. So this is yogurt. And then if we use probiotic, so the peptides below 10 kilodalton molecular weights, this is the contribute for the AC inhibitor. Yeah, this is the proven result. The next one is cholesterol assimilation of yogurt. So what is this? What the table is? Yeah, the table is explain the types of yogurt: yogurt Rossel, probiotics plantarum, yogurt Rossel probiotic fermentum. That Lactobacillus fermentum and Lactobacillus plantarum has gene DNA to produce cholesterol assimilation agent. Yeah, so we only use two probiotic plantarum and fermentum. So the data here, yeah, we can see that yogurt probiotic with Lactobacillus plantarum has higher cholesterol assimilation compared to yogurt and yogurt Rossel. Also, the yogurt Rossel with fermentum, yeah. So this is the proof that specific strain of Lactobacillus plantarum and fermentum can give more or higher, yeah, cholesterol assimilation function of the yogurt. Okay, the next one. Why it cannot move? Why it cannot? Yeah. Okay, I stop share and I will give to participants to give a question maybe, yeah. Okay.
Okay, thank you, Prof. Irma. Maybe we have a lot of questions at the chat room, but for the first, I want to offer the participant directly to ask Prof. Irma. Maybe you can use raise hand feature, please, if you want to ask directly. Or still waiting, we can read the question first from, I want to share the file.
I have 89 slides. And now I only explain to the 36 slides.
Yeah, we have a lot of questions, Prof. For the first questions from Rafida from Malaysia. Assalamualaikum, I'm Rafida from Malaysia. It is possible for the good bacteria or probiotics to be harmful bacteria due to any mutation. Is it possible, Prof?
Maybe. So we can maintain the quality of the probiotic in our laboratory. It should be maintained. Yeah. For example, if we use only one culture from one stock, yeah, and then we use it up to until maybe 5 years, yeah, it will be changed. Maybe it will be changed before the little trees not maintain the quality, yeah. So we can kill it, yeah. We can kill the bacteria, and then we can use from the first stock, so we can maintain the quality of bacteria.
Yeah, it is an important thing to maintain the bacteria, Prof. And then
Yeah.
the second question from Dian Nur Amalia from Indonesia. I want to know how about fermented sauces made in Indonesia that we should test the organoleptic properties. So it is safe
Yeah.
to eat it without
Safe.
cooking first. It's very
Yeah, safe.
because it's a fermented food, right? It's safe for our body. And then the next question from Bristi, Bangladesh. I want to ask, what is the difference between probiotic and prebiotics? What is the difference, Prof?
Probiotic is life microorganism. That prebiotic is carbohydrates for the food of the probiotics. So the prebiotics is food for probiotics. Sometimes for example, carbohydrates. Yeah.
Okay, it's a good explanation. And then from Ganda Adi, Indonesia. Assalamualaikum, Prof. I'm from Indonesia. I want to know, can the good bacteria from the fermentation process of a product survive and work optimally at a low pH in the stomach?
Yes.
It has GERD, yeah, especially as
Yeah. So you can you also consume it after you eat. Yeah. Better after you eat. After you eat, you can consume the yogurt or etc.
because when the stomach is empty, it's very lower than after you eat it. And then the questions from Trust Ambora, Indonesia. I want to ask, why do people consume fermented products that can cause indigestion? For example, yogurt and kefir. Why? Even though in yogurt they are good bacteria for digestion.
I don't know.
Did you get the point, Prof?
No, because if I cannot catch here the question. Yeah, and we are health, so we can consume it the sour products or fermented products. But if we unhealthy, please be careful. Yeah, after eat, then we can after eating you can consume the probiotics. Yeah.
Okay. Well, maybe Trust Ambora, can you open your mic and directly ask to Prof. Irma? Is it possible? Where is you? Where are you? Okay. Maybe next time. Maybe. So the next question from Desti, Indonesia. Why in the fermentation process the product should not be exposed to light, and why it's called an anaerobic process?
Why the fermentation product? No. Anaerobic process is no need oxygen. Yeah, not depend on the light. Yeah.
Okay. Just when the fermentation process we keep to prevent from light. Yes. And then from Ranti, Indonesia. How do we know the food is already in the fermentation safe to eat?
Yeah. It's need specific skill, yeah, maybe, yeah. But so is the lactic acid taste is very specific, yeah. Not alcoholic fermentation. So if yogurt, you produce yogurt, but with two bacteria, Lactobacillus bulgaricus and Streptococcus thermophilus, and then the final products, you see the alcoholic in this product, so it is not safe because it's contaminated with another bacteria. Because the Lactobacillus bulgaricus and Streptococcus thermophilus is homofermentative bacteria. For example, like that, yeah.
Okay, next explanation, and then for the next question is from Dita Indonesia. What are the factors that fail to make kefir and yogurt? What is factor affect the kefir and yogurt process?
Yeah, please pasteurize fresh milk, yeah, before using the milk to produce the kefir and yogurt. And then from the for yogurt, please use the starter culture, yeah, Lactobacillus bulgaricus and Streptococcus thermophilus. And from kefir, you can refresh before, yeah, you use it, yeah. Refresh. You can ferment the kefir grains in the milk, and then you can fresh one of the kefir grains and then use it. Yeah, I think it will be safe. Yeah.
Okay, thank you, Prof. And last question from Rafila, Indonesia. As we know that there are several beauty masks here that use the main ingredient by kefir. I want to know something kefir that can be consumed.
Yeah.
The same as that. It's confirmed.
Confirm.
Can be used as a mask.
For the beauty mask. Yeah. It is safe.
Okay. Any questions from the party?
Okay, maybe I will continue. Yeah.
Okay.
This is very specific. Yeah.
Yep.
Can you catch my slides?
Perfect, Prof.
Okay. Yeah. I will explain maybe fastly, yeah. More fastly, yeah, fastly. Okay, this is the mechanism of the hypocholesterol, yeah. I send the slide to the Prof. Asep, yeah. Maybe it will distribute, yeah, into the participant, yeah. So this is the mechanism of hypocholesterol system of the fatty acid bacteria, which by short hydrolysis enzyme. So the next one, no. I have difficulty, yeah, into move to the. So maybe here, yeah. I'm so sorry, yeah. Okay, and the next one, it's the data about the anti-diabetic functional properties, yeah, of yogurt with roselle, with probiotic and roselle, yeah. We analyze enzyme alpha glucosidase inhibition of yogurt. We compare yogurt and then yogurt probiotics, compare then yogurt probiotic roselle. So, what is the enzyme alpha glucosidase? Alpha glucosidase is an enzyme that plays an important role in the absorption of glucose in GI tract. So, if we can inhibit this enzyme, so the absorption of glucose in our intestine will decrease. So it will be beneficial for diabetic patients. Yeah. No. Okay. So let's see. Yeah. Let's see in storage control. Yeah. Before the storage. Yeah. So yogurt probiotic and egg with roselle has higher activity of enzyme alpha glucosidase inhibition compare yogurt and yogurt probiotic. So very symbiosis mutualism, yeah, between probiotic and Rosel. So now we can use, so now we can commercialize it, yeah, yogurt probiotic with Rosel extract, because it has many beneficial functions that has been proved in our research. The next one is the mechanism, yeah, because the fermentation in our intestine by probiotic will produce short chain fatty acid, and then short chain fatty acid will block the insulin and so on. Okay, this is the new research, the new result. I explain because it has been published, yeah. In vitro anti-cancer potential of Lactobacillus plantarum 185 and Lactobacillus acidophilus 2b4 extracts against human colon cancer cell lines. Yeah, we know that colorectal cancer is an emerging public health problem in Indonesia. Yeah, so it will be challenging, yeah, to prove that our probiotics can contribute the inhibition of the colon cancer cell lines. This is the result. We proved it by some bacteria, yeah, some intracellular and extracellular from the bacteria attached to the colorectal cell line, yeah. This is the result. Nice. This one, yeah. This one is control. Control is cancer cell line, yeah. The light one is live cell lines, and the second one doxorubicin. Doxorubicin is positive control. Positive control doxorubicin is medicine, yeah, for the cancer. So the black pool is the cancer cell that die with the doxorubicin. The another one, Lactobacillus plantarum intracellular substrate in the inner, yeah, intracellular, and extracellular substance that extracted into the extracellular out of the inner, yeah, cell. So we can get the intracellular is better than extracellular. So this is important to consume live microorganism, because the live microorganism, so we can consume all the bacteria, yeah, and the substance can get the cancer cell lines is inner the cell of bacteria, yeah. So we can consume the live microorganism. Okay, this is the another one. We can publish. We publish it into the Journal of Food Science, Q1, yeah, of journal, yeah. We explore about the fermented sausage. We used beef, yeah, for the source of the materials. What a background of the products. Rutan Esbalinis traditional fermented sausage, which is made from lean pork, yeah, fat mixed with spices, sugar, and salt, and then stuffed into clean pig intestine, and then fermented under uncontrolled conditioning. High protein content as compared to the breast meat. And then this is the type of the urutan, spontaneous, uncontrolled microbial treatments that we can make new products, new type of the fermented sausage with the Lactobacillus. So Lactobacillus can be applied to control the quality of the product. Lactobacillus are responsible for lactase production, for the flavor of sausage, contribute to the nutritional value that are produced during fermentation. What is the beneficial of fermented food? Inhibition of intestinal pathogenic bacteria, prevention of pathogens, production of bacteriocin, and increase of food assimilation. This is the research, better profile of monounsaturated fatty acid and polyunsaturated fatty acid, longer cell life at room temperature. So probiotic addition could increase the quality of traditional fermented sausage. This is the other one. I think this one in Netherlands, yeah, and the right one in the Adelaide, Australia. So, but I didn't eat it, yeah, because it comes from pork. Okay. So in the middle of fermented our slides, yeah, the next topic about the bacteriocin, maybe I can explain fast, yeah. So what time is it?
I only 5 minutes remaining, bro.
Okay, I only have 5 minutes, yeah. So everybody, if you interested in our topic, yeah, please, yeah, please register in the next semester, yeah, with our topic, yeah, in microbial diversity, yeah, in bachelor program and applied microbiology in master program. I have 2 courses, yeah. Next semester we'll explore about the beneficial bacteria. Okay, this is the bacteriocin. I will move it, yeah. The bacteriocin natural preservative can be isolated, yeah, from the Lactobacillus. And then this is one, the mechanism of the bacteriocin. This is our paper. It has been published in Beneficial Microbes 2015 in Q2, yeah, Q2 journal, and Arifin also, yeah, has publication in QT, yeah, with peptide journal, very specific journal. We can explore about the gene in the bacteria, and then how to isolate, yeah, the protein from the bacteria, and then we can get the single band of protein. This is the SPIC. This is a mechanism, yeah. We can select the treatment to explain the mechanism of the antibactericidal, yeah, how to kill the bacteria. Okay, this is the scanning electron microscope, the leakage of bacteria. Okay, this is the picture of our bacteriocin. Okay, this is the procedure of making fermented sausage, yeah. And then this is the research to prove that our Lactobacillus plantarum is in the fermented sausage. Yeah, we explore the biomolecular identification of specific gene. This is another one. Okay, this is the PLNEF, plantaricid EF, yeah. Okay, this is the next one, non-fermented foods. The second topics, yeah, 5 minutes, yeah, or 4 minutes. well flat, so high biological value protein. We need animal products, important micronutrients essential for good health. For example, the other non-fermented food, traditional fermented food in West Sumatra, again, we can find dendeng batokok from Jambi, Sumatra, Bengkulu, yeah. Sei is traditional smoked beef from East Nusa Tenggara Province, shredded, yeah, or abon in Bahasa. Another one, rendang, yeah, special meat products, yeah, originating from the West Sumatra, Padang. Another one is egg, yeah, salted egg, yeah. The other one, pindang eggs. Oh, this is honey, yeah. Why honey? Because Indonesia has many many specific of this. Yeah. This is our our research, yeah, and meta-analysis of honey with Ibu Astari also, yeah. The study evaluate effective the propolis extract of natural honey, yeah. This is the one table to explain, yeah, the output, yeah, of inhibition of the honey to the pathogenic bacteria. Okay, this is the composition of nutrient of honey. Okay, thank you very much for your attention. Thank you, Pak Arifin.
Okay, thank you so much, Prof, for your sharing knowledge and very, very interested, especially fermented product from animal product. It looked like very delicious for me. And then I offer to participant again. Any questions to Bu Irma? Please, you can raise hand or you can write at the chat box. Any questions? I have okay. Kalisa, okay, you can open your mic.
Okay, before I want to thank to Prof. Irma to the presentation, and maybe if you don't mind, I would like to ask the question that related to previous presentation. May I?
Yeah.
Okay, thank you, Prof. As far as I know, cheese that sell in Indonesia is not made by originally fermentation process, but it is made by cheese that ready consumption. Like they have cheddar cheese as their main ingredient. What I want to ask is the Indonesian market cheese still have the good function to our body compared to fresh cheese that originally made fermentation process? And the second question, maybe as you mentioned before, that yogurt with Rosella added, is there amount limit of Rosella that can be added to the yogurt probiotic? Maybe that's all I can ask. Thank you, Prof.
Yeah, thank you, Kalisa. It's right, yeah, that cheddar cheese is not fermented cheese, yeah. Also, what is the cheese from for the pizza, Mas Arifin? Cheddar cheese. What type cheese for the pizza?
Mozzarella.
Okay, mozzarella. Mozzarella also, yeah, not fermented process, yeah. So it different, yeah. And Indonesian people likes not fermented cheese, yeah, compared than fermented cheese, because the fermented cheese like maybe specific taste, also specific flavor. that Indonesian people don't like it. Yeah. This is the answer for the first question. And the second question, you asked about what?
The amount limit of Rosella that can be added to yogurt probiotic. Is there any limit?
What percentage, Mas Arifin? I forget. 5%?
Yeah, approximately 5 until 10%.
5% until 10% with naturally extract of Rosella flower.
Thank you for the answer, Prof. Irma.
Okay.
Okay, anyone else? Or I want to ask to you, Prof. I'm very curious about the test of Dadia as your breakfast today. Can you describe the test, Prof? And why until now the Dadia cannot sell in the modern market, especially? What is the challenges to scale up this product?
Yeah, because the texture is solid, yeah. So the buffalo milk maybe has the higher lipid, yeah, or fat content. So the final product is very solid. And then very sour. I think the H value is about 4, yeah, 4 or below than 4, yeah, maybe. And then this is in the bamboo, so it's very limited. Not a good presentation, yeah, in the bamboo, yeah. And voluminous, yeah. And then if you bring it to the plane, yeah, to the plane, it will give very little difficulties, yeah, to throw the X-ray, maybe the first one, yeah, because it's from bamboo, yeah. And then second one, the naturally bacteria in the bamboo give the specific taste, yeah, for the milk, yeah. So if we change bamboo with another package, yeah, with another package, maybe from plastic package, so it cannot, maybe it will different. It will different, yeah, different taste, different flavor, yeah. I think so it's very traditional fermented food, yeah. Only two days in the room temperature. It can maintain the quality.
Can we isolate the bacteria from bamboo, Prof?
Yeah, maybe. But what for? But what for? Yeah.
Different taste from originally product, maybe.
Yeah. But what for? Yeah. If we isolate latency bacteria from the daddy, so the latency bacteria maybe come from the bamboo and go well in the milk, yeah. So it can be used for another product, I think.
Okay, thank you, Prof. Maybe it's enough for this class. I want to thank from Prof. Irma. I hope you're still with us for a while to submit the certificate from Bu Tuti as the head of Animal Production and Technology Department. Bu Tuti, are you there?
Okay, I'm now.
Yes, please, Bu Tuti.
Okay, Professor Irma, thank you for your very interesting presentation, and thank you also for your support to the DAPT Summer Course Program. Please accept this certificate as an expression of our gratitude and appreciation for sharing the knowledge you have provided.
Thank Thank you, Bu Tuti. You are welcome. Thank you, Mas Arifin.
Thank you so much, Prof.
Also, thank you for the all participants. Yeah.
Thank you. I'm sorry to disrupt your time, especially on weekend.
No, it's no problem.
Thank you, Bu
Okay,
Tuti.
thank you. Thank you, Bu Tuti. Thank you, Mas Arifin. So, can I leave now?
Yeah. Thank you, Prof.
Okay, bye-bye, all participants. Assalamualaikum.
Waalaikumsalam.
Waalaikumsalam warahmatullah.
And the next session, the committee will be have a. Oh, sorry. Oh. I forgot to take a picture together with Prof. Irma. Maybe next time. Thank you, Bu Tuti. The committee will play a video, 2 videos for participants. Terima kasih, Mas.
Like the ones found in this 15th century palace of Cherabach. Take the train, let it take you back in time, and enjoy the timeless feel of a magic ride. Here, every tale is expressed by genuine human relationship, and by the deft touch and tender care of artistry. The great beauty of batik has a life of its own in this land. Time-honored values are preserved in body and soul. This is where a masked dance brings us wisdom about life. And a goat fighting demonstrates the meaning of survival. Bamboo has a special place in everyone's heart. Its melody is nothing but an infectious charm. The charm that echoes throughout the world. The plant is everywhere. It is a part of life. It unites the people with the mother of nature. In a sense, bamboo has become the epitome of harmony. Meanwhile, in the South Coast, witness the insistence of a sustainable life, a life reserved for the generations to come. In this wonderful hidden paradise, if you love water adventures, you're in heaven. From the ocean up to the river, nothing is off limits. Adrenaline rush knows no boundaries. It gives you the moments to live for. Just outside the city, there's a collection of wild animals from all over the world. Social understanding is developed. Trust is established. In transformed. In the capital, tradition comes in style. Urban festivals frequently grace the streets. Local dishes and international menus are presented. In the warmth and friendly manner Bandung is known for. Fashion and lifestyle have long been the synonym of the city. It is a shopping nirvana. The lush and beautiful landscape offers only the best of serenity. From the five-star hotel's luxury to the intimate lodge's tranquility. You can always take a journey to the mountain and walk down to the crater. It also has the most extensive botanical garden in the country. It's a perfect choice whenever you want a place of solitude. Your time and effort will be immensely rewarded by the spectacular beauty of West Java. Wonderful Indonesia.
A wondrous civilization. Disposed to tranquility. With scenic landscapes bordered by the sea. Here, mythologies become principle. And religions create the cornerstones of life. It is contained in the arts and crafts, where celestial ceremonies become part of popular culture. Legacies of glorious traditions, where folktales are regarded as guidebooks to perfection, honed over centuries. Yet possible to experience within a few hours. Experiences you can take home. Here, art preservation is a tourist attraction. History is a living monument. Even shopping is meaningful. See world-famous resorts compete with local means of accommodation. Enjoy natural kindness. Or the excitement of exploring nature. endless stories to tell, and never-ending feasts. Never-ending possibilities to get more out of life and journeys. Yogyakarta. The mountainous region of East Java. Where Majapahit once stood. Largest ancient kingdom. Changing and opening slowly over hundreds of years. A historical city on the move. A celebration of remarkable heritage, grandeur of the past, wondrous ancient architecture, and the outstanding universal values behind, where art absorbs the audience into colorful drama. Still, the exotic mesmerizes. Extraordinary feasts exhilarate. Bring us back to the land of the ancient mountains. A myriad of outdoor adventures. In exceptional panoramic nature. Exciting variety. This is a land of historical significance, where the old days are celebrated. Local cuisine is cherished. Beyond the city. Preparing for a thrilling ride on the vast sea of sand in Bromo, East Java.
Thank you, committee. For the participant, we will start again at 3:15. So you can take a break first. And see you for the next class. Thank you.
Hi, good afternoon, everyone. Can you hear my voice?
Yes, clearly, Bu Desi.
Okay, perfect. Okay, so I hope that everyone is already get back from your break time, and we're going to start our next lecture. And we have Dr. Hugh Lin Huang from National Chung Sing University of Taiwan. Hi, Professor.
Hello.
Hi. Are you there already? Great. Thank you. Hey, good afternoon, and thank you for being here with us for this summer course program. It's really nice to have you here today in this your weekend, but you still have sometimes to be here with us. So we really appreciate that one. And let me read the curriculum vitae of how should I call you, Dr. Hugh Lin?
Huang.
Sorry.
Xiao Lin Huang.
Xiao Lin Huang. Okay,
Yes.
great, perfect. So Dr. Xiao Lin Huang is associate professor at the National Chung Sing University of Taiwan. Am I correctly spelling it?
Yes.
Okay, perfect. And she is a lecturer. Mainly, she teach about the animal genetic breeding and animal molecular biology. And she has many collaborations, especially related with this aspect, such as collaboration with Livestock Research Institute, Council of Agriculture, and also she also have a collaboration with the Department of Medical Laboratory Science and Biotechnology, Kaohsiung Medical University, and also National Taiwan University and National Museum of Natural Science. And today she's going to explain us, and it's really interesting topic, of course, and it's about searching for new molecular markers. Yeah. Oh yeah, I forget to introduce myself. So I'm Windy. I'm a lecturer at the Faculty of Animal Science, Bogor Agriculture University. And today I'm going to be your moderator. And we have in total, we have 50 minutes for total lecture. And I suggest to have like 30 minutes to 40 minutes for lecture and like 10 minutes of discussions. So, Dr. Xiao Lin Huang, the time is yours.
Okay. Okay. Thank you, moderator. As you mentioned, my topic is searching for new molecular markers. So why it is important? Because modern strategies for animal breeding is molecular breeding. Molecular breeding is not a new term. Actually, it is an international journal. The fact is, our lab helped animal farm to establish molecular breeding center. Related to animal breeding, it leads to successful husbandry. Because traditional animal breeding, for example, progeny test, it is time-consuming and costly. It is a new trend for gene markers to predict adulthood performance of young animal at early age to reduce feeding and management cost. So those are SCI papers we published. The strategy for us to search for new selection markers are we use oligonucleotide or cDNA microarray as probe because on the microarray, those sequences are known. And we use targets from groups with high or low performance labeled with fluorescence, and then search for significant different expressed genes, followed by DNA automatic sequencing and genotyping, and finally for statistic analysis. Of course, we do candidate gene identification. So one of the experimental animal we used is egg duck. Egg duck is one of the major egg-laying ducks bred in Taiwan. It is also a maternal line of major meat duck breed. And that is the result that we collaborate with Livestock Research Institute. And this is the picture of meat duck. In our study, we used all female ducks. We use artificial insemination, AI, not artificial intelligence. So those female ducks, artificial insemination with mixed salmon. Why we use artificial insemination? Because egg duck and meat duck, their body shape differs. There was no nature mating, so we use artificial insemination. And those are important reproductive traits that we record. One is fertilization rate, and the second is hatchability. Finally is fertile period. The definition of fertilization rate in our study, the ratio of fertilized eggs to collected total eggs. Under the light, as you can see by candling, you can see embryo and vessels inside the fertilized egg. For higher fertilization rate, it means better return on investment regardless laying or meat ducks. As for hatchability, this is the ratio of successful hatchings to fertilized total eggs. And because sperm into female reproductive tract cannot survive for very long, about several days, so the duration during the fertilized eggs were continuously observed is called fertile period. We use microarray chip technology to find new genes. In our study, we use Cy5 or Cy3 fluorescence to label high-performance animals or low-performance animals. Because the affinity of RNA and cDNA to fluorescence, that's why we did dice web experiment, and the results were similar. And this is the first part result. Use this genome-wide transcript expression analysis. We focus on several important genes that is differentially expressed between high and low performance dogs. One of the important genes is TMIGD1. TMIGD1 recognized as an adhesion molecule because it contains two putative immunoglobulin domains, and it enhances cell-cell adhesion by increasing transsubstantial electrical resistance. This is why we expected that TMIGD1 uses barrier function to adjust cell permeability. To link cell permeability with sperm activity, because several important factors related to sperm activity can be affected by cell permeability. For example, lactic acid. It maintains sperm survival in female duck tract. And also fatty acids and HISHA proteins. They reactivate sperm from stable state and for further fertilization. So we suggest that TMIGD1 use its barrier function and to adjust cell permeability, release factors that maintain or reactivate factors sperm activity and change fertilization rate. And the second important gene is OTX2. It is lethal if one knock out OTX2 gene. Destroy downstream gene of OTX2 leads to impaired fertility. We use real-time PCR to confirm the result from microarray. So here you can see real-time PCR result and microarray result all shows that gene with similar expression pattern. And we also checked protein expression by Western blot. For preliminary comparison, you can see here is a polymorphism G175 G to A single nucleotide polymorphism. For DNA sequencing result, there are 3 genotype stocks: GG genotype, AA genotype, and GA heterologous stocks. We use FAM and BIC to label different probes for genotyping. Use this method can search more than hundreds of dogs within 40 minutes. So we called high throughput screening. And this is the high throughput genotype screening result. We use BIC to label probes with A and use FAM to label probe with G fragment. And then here you can see 3 genotypes. The data were confirmed by mini-sequencing. Why it is called mini-sequencing? Because it only extends 1 nucleotide. So here you can see 3 genotypes under genotyping technology. For PCR reflip result, reflip stands for recursion enzyme length polymorphism. So under digestion of RSA1 recursion enzyme, GG genotype fragment is contact. However, AA fragment were digested into 135, 83 2 fragments. The heterozygous ducts, you can see the combined pattern. So it is very cheap and quick result to analyze genotype. For genotype and phenotype statistic analysis, we can see GG genotype dogs with the highest fertilization rate compared with other genotypes. However, for fertile period and hatchability, there are no significant difference among genotypes. For the hatchability experiment, it is a fact that concentration of minerals in egg or age of poultry or egg sequence position may affect hatchability. However, we are interested in molecular biology study. So in poultry oviduct from infundibulum, magnum, isthmus, uterus, and vagina, we focus on magnum to see expressed genes
of egg white proteins. Egg white proteins, for example, ovomucoid, riboflavin binding protein, transport into embryo during embryogenesis. So those egg white proteins, for example, ovomucoid, ovobumin, cannabulin, lysozyme, riboflavin binding proteins. When those proteins with mutations, it affects hatchability. So we use cDNA from RNA and fabricate on chip as probes with known sequences. And for high or low hatchability groups, we use also Cy5 or Cy3 to label, and after hybridization, image was captured and for analysis. And this is the fabrication process. And this is the hybridization process. On the glass, probes are on it and target hybridized with probes. So Cy5, you can see red color label with high performance group. Cy3, green color label with low performance group. In this experiment, high hatchability is 88%, and low hatchability is about 55%. If we use fold change 1 as criteria, there are 17 upregulated genes and 10 downregulated genes. Change criteria to fold change 1.5. There are 3 upregulated genes and 3 downregulated genes. One of the genes is ovomucoid. We also use real-time PCR and Western blot to check, validate the result. So to compare microarray, real-time, and Western blot result for egg white protein, ovomucoid O expressed more in low hatchability group. This may indicate that ovomucoid overexpression may not good for hatching. Ovomucoid gene express 19% mRNA in oviduct. It is well-known protease inhibitor with highly conserved amino acid sequences, and its physiological role not yet understood. It flows into poultry embryo during embryogenesis. Because ovomucoid is a protease inhibitor, its substrate subtilising, knocking out, leads to embryonic lethal. So according to ovomucoid sequences, we use forward and reverse primer to amplify this specific area for PCR reflip. And we found that there is a deletion in the fragment in low hatchability ducts. And this deletion creates a tau domain protein binding site AATTC that is not in intact fragment. So here you can see with PCR reflip result, minus indicate a fragment with deletion, and positive indicate the intact fragment, and heterozygote fragment contain both. So tau domain protein binding site were found by signal scan software. Tau domain protein binding site is highly conserved among different species, and it is a transcription factor binding site, regulate organ development and cellular differentiation. And the deletion created tau domain protein binding sites may increase ovomucoid transcription level and reduce hatchability. For different genotypes of ovomucoid, here we can see positive ducts with the highest hatchability. However, negative with deletion ducts, homozygous with the lowest hatchability. So normally we will eliminate minus ducts. However, heterozygous ducts may also produce minus offspring. So if possible, we will also eliminate heterozygous ducts. To summarize ovomucoid experiment, both mRNA and protein level of ovomucoid higher in low hatchability group indicates that high expression level of ovomucoid may not good to hatching. And for deletion created tau domain binding site increases transcription level of ovomucoid, and maybe that is over beyond the needs, so it may reduce hatchability. A novel PCR reflip marker of ovomucoid can be used as genetic marker for marker-assisted selection to improve hatchability in Cai ducts, that is the egg duct I mentioned. For fertile period, here you can see after artificial insemination, 14 days still have sperm in female reproductive tracts. So sperm will hide in uterovaginal UV junction and maintain their wait for fertilization. To prevent sperm precursors activation, so sperm stay in UVJ before fertilization. So here is sperm storage tube in UVJ. Here you can see a contact of sperm with epithelial in female reproductive tracts. So we also use microarray to do this experiment. With 3 independent biological repeats, you can see use same crop and same targets in different chip, repeat 3 times. The results were similar. And those are genes we found in this study. And the reproducibility is very high. For those genes we found in this study, we further use CAG to analyze pathway. And we found TVO gene is very important in this study because it is required for dynamic acting reorganization of T cell activation and immune reaction. So it is speculated that female parent birds in short period ducks could be allergic to the incoming sperm and take sperm as an outsider. And TVL gene overexpressed, and then sperm may not stay in UV junction for a long period of time. And this is mRNA and protein level validation result. And for NPY gene, it decreased in blood vessel and supply between blood capillaries and local tissue. So NPY upregulated in short period ducts. The subepithelial blood vessels of sperm retriever in UVJ in short period of ducts increasingly expressed NPY, decrease the blood supply, and decrease GAG level. It causes precocious sperm activity and fertile in ducks. For TRAK1 proteins, it is target OGT2 transcription factor. TRAK1 mediated transcription inactivation may prolong fertile life of sperm in UVJ and extend fertile period. To summarize this experiment, MPY and EBL were significantly expressed at transcription level in short period of group. Because MPY may reduce substance supply to local tissues. For EBL, it is important in immune response of T cells. Furthermore, TRAK1 was significantly expressed at transcription level in long period groups. It mediates transcription inactivation. It benefits for prolongation of lifespan. Although interpretation of results in the study is limited, more survey will need to determine physiological functions of these genes. Our results provide evidence that gene expression of MPY, EBL, and TRAK1 are associated with length of fertile period. We also did some experiments in pigs. Those are markers associated with pigs for meat quality genes. For example, fatty acid binding protein. For reproduction, those genes like estrogen receptor, prolactin receptor. And for growth of pigs, MC4R is also an important gene. For live-born litter size of pig.
Excuse me,
Yes?
Dr. Xiulin Huang. We
Yeah?
have 5 minutes for presentation and 10 minutes for discussions.
Okay, so I have several, this last slide.
Yeah, okay.
Okay, and for winged litter size, no significant difference between two genotypes. For AC2 genes, there are significant difference between genotype for live-born litter size and winged litter size. Okay, thank you.
Perfect. Thank you very much, Dr. Xiulin Huang.
Huang.
Sorry?
Xiulin Huang.
Xiulin Huang. Yeah. Oh, sorry. Thank you for your presentation. It's really nice. And we already have some questions here. Shall I read it for you? Okay. So we have questions from first Professor Jakaria from IPB University. And thank you, of course, for your very interesting topic. And I have two questions. Can the SNPs found be used as genetic markers for hatchability in ducks? And what do you suggest about the application of marker-assisted selection in livestock productions?
Okay. So here is PCR reflip molecular marker for hatchability. So for minus ducks, it reveals the lowest hatchability compared with positive ducks. So if use ovomucoid PCR reflip marker, you can use for marker selected selection. So here you can see for hatchability, you can use this marker. Okay, so the next question is, yes, it is okay for use for marker selected selection.
Yeah. Is it answering the question, Professor Jakaria?
Okay. Thank you very much.
Yeah.
Thank you.
Do you have comment?
Okay.
Okay. Thank you very much. And for the next question, there is a question from Malaysia. Rafida. Hi, Rafida. Her question is, for any polymorphism that have been observed, how does the polymorphism affect the phenotype? Do the SNPs that have been found alter the amino acid sequences?
Well, yes. Actually, here the same experiment, you can see the polymorphism can affect phenotype, because polymorphism is minus or positive or positive minus. And the phenotype is hatchability, fertile period, fertility, or egg weight, for example. And some SNPs can alter amino acid sequence, but we did not show here. So actually, yes, SNP can alter amino acid sequence, yes.
Okay, I think it's quite clear. We still have like 7 minutes for questions or discussion or anything that is not clear, or do you want to ask more? Feel free. Otherwise, I also have a question. Actually, not really related with this genetic, because I'm not in the genetic field, but I do really interest with this the type of ducts that you use as your experiment. So you use I don't know how to spell it correctly. Maybe Saya.
Yeah, Saya duck.
Saya duck. Is it kind of typical duck in Taiwan, or is it like the original breed, or is kind of what is that? I mean, is that the really dominant breed in Taiwan? So you use this one for your research?
Actually, it is original from Taiwan, and it is a major egg duck. So it produces egg. Normally, it can produce more than 300 eggs for a year. So it is very good to use Saya duck.
Okay. Wow, very interesting. And would it be also because you also compare different hatchability between the treatment? Would it be possible also to do the same method for other, for example, other ducks or other types? And would you also expect the same result as you have right now?
Yes, if we use meat duck, we will focus on genes related to meat production. And for similar technology, we also can use for other animals. For example, use for pigs, for cow, for example. So it is also can use for different dogs and also for different animals.
Yeah. Okay. Wow. It's quite clear, I think. Are there any more questions from participants for this lecture? I think it's really interesting. Yeah, we study. We know that how the genotyping and also new marker that you are trying to use in your field, especially in the ducks in Taiwan. So that's really interesting. We also like to eat like Taiwan duck. I think it's quite famous in Indonesia. So I'm wondering whether this is also from this breed or not. Okay. Oh yeah, there is a question. There's one. I think it's the last question. It's from South Korea, from Venuste Chungnam from Chungnam National University. Sorry. How can we minimize the inhibitance of ophthalmocotes to maximize the hatchability? Yes, how we can minimize these inhabitants?
Yes, because higher ovomucoid expression is not good for hatching. That's why we eliminate minus ducts, because minus ducts produce more ovomucoid proteins. So we eliminate minus duct. And we also want to eliminate positive minus ducts because the offspring may also produce minus offspring. So if we eliminate those ducts and collect positive ducts, and I think it will be a way to improve hatchability.
Okay, but I think it also make me curious. How would you exclude this one? Is it like you just select it for the ducks, for example, that only have the positive one and just directly remove it, or is there any more criteria for that one? Because you said that having more ophthalmocoids would limit the hatchability or reducing the hatchability. How would you eliminate that one?
Because if we have genomic DNA from those ducks, we can identify the genotype. So normally we draw blood from ducks at any age, and especially you can eliminate at very young age. So if we eliminate minus ducts, because we check blood genotype, the genomic DNA. So we eliminate positive minus and minus ducts at early age and maintain positive ducts. So we can save cost to feed them further to adulthood. So you just need to withdraw blood and collect DNA from those blood, and then do genotyping.
Okay. Oh, well, yeah, I think it's quite clear. Thank you very much for your explanation. And I think the time is up. And I would like to thank for your lecture for today. It was really nice. And for me, it's really interesting because I'm not in the field of genetic, of course, but this is quite interesting because I know that there are a lot of things that's going on related with this genotyping and finding new markers. So really nice. Thank you very much. And I would like to invite Professor Jakaria to give a certificate for you. Professor Jakaria is also in the field of genetics, so he must be really excited with your course. And yeah, Prof. Jakaria, the time is yours to give the certificate.
Hi. Thank you, Dr. Windy Azahra. Good afternoon, everyone. On behalf of the organizing committee, we would like to give a certificate of appreciation to Associate Professor Xiulin Huang for significant contribution in summer class today. Thank you very much.
Yeah, thank you very much, Professor Jakaria. Maybe we can have a picture together. Komita, can you help us to take a picture together with our speaker? Yes. For everyone, please turn on your camera. Wait a minute, we lost our speaker, Dr. Xu. Are you still there? Okay, you're back. Okay, perfect. You're back. We're going to have a photo session for now, and for everyone, I hope that you can turn the camera on so we can have a nice photo session now. Komita, can you help us to take a picture, Mas Arifin?
Okay, Bu Windy, let's prepare yourself. I have 4 screen on my device.
Yes. Okay. Smile, everyone.
1, 2, 3. Okay, the second one. 1, 2, 3. The next. 1, 2, 3. The last screen. 1, 2, 3. Okay, thank you.
Okay.
Thank you.
Well, thank you very much.
Thank you very much.
Yeah, thank you very much for your presence and also for your lecture. It's really appreciated, especially during this weekend time. Really much appreciated for your time and also your knowledge. That's really beneficial for us. And we'll continue for the next lecture in 5 minutes. So we are going to prepare a lecture from Pak Cahyo. Pak Dr. Cahyo is a lecturer from IPB University, and then it will be moderated by Bapak Arifin. So stay tuned in this Zoom meeting for the next 5 minutes. This is the last session for today's event. So thank you very much, everyone, and I'll see you again in a couple of minutes. Thank you.
Hello, the committee. Can we start now? Okay.
Yes, please.
Okay. Thank you. Hello, everyone. Welcome back to our last class for today. Before we start today, I remind you again, all of the participants, please turn on your camera. And then if you have any questions or comments, please use the raise hand feature to talk directly to our lecturer or write your question or comment at the chat room. I hope you guys are still excited for this class because our lecturer for this session is very expert especially in the field of protein engineering, and namely Dr. Cahyo Budiman. Pak Cahyo, are you there?
Yeah, I'm here.
Okay, thank you, Pak Cahyo. Thank you for your attendance in our class, and sorry to disrupt your weekend. It is an honor for me because Pak Cahyo, my professor too, with Bu Irma, when I studied master program a few years ago. First of all, let me introduce Pak Cahyo, this committee. Okay, thank you. Pak Cahyo Budiman, PhD, took his postdoctoral research at Okinawa Institute of Science and Technology, Japan, with the field structural biology, and then his PhD and master program at Frontier Biotechnology, Osaka University, Japan, with field protein science engineering, and his bachelor program at Animal Product Technology, Faculty of Animal Science, IPB University, with final year's project with milk fermentation. Okay, please. Pak Cahyo also joined several organizations such as Protein Society, Protein Society, Biochemical Society, Japan Protein Society, and then the ASEAN Federation of Biotechnology. And his research interest and publication is enzymology, protein engineering, molecular biotechnology. Next, please. He has a 10.4 Scopus high index. It's come from 320 to 20 citation in 49 documents, and he also have some awards such as CEO Summer Course Fellowship, and then Max Scholarship Japan, Research Fellowship, Visiting Researchers Max Planck Research Unit, Enzymology and Protein Folding, and then a lot of awards, I think. So for the next step is Pak Cahyo, I will remind you about the time of presentation is 40 minutes, and for discussion is 10 minutes. Okay, next, Pak Cahyo, please to deliver the lecture material. The screen is yours.
Okay, thank you, Pak Arifin, for the kind introductions. I'm looking for my eyeglasses, cannot see clearly now. All right, okay. Let me share my screen first. Can you see my slide?
Yes, perfect, sir.
Is it in the full slide?
Yep.
All right, okay. Good evening, everyone. For last lecture for today, I hope my lecture will be interesting enough to avoid your to fall to sleep during this sleepy times. All right, I would like to deliver the talk on animal-based proteins for fighting the viruses. Unlike to the other lecturers before, that they talk based on the extensive works on the topics, I would like to, in my talk, it is purely just lecture, so it is based on the previous research done by other people. Then maybe I will add on my own experience on the related topics. All right, let me start with showing you some figures here. I believe everyone knows what it is, milk and dairy products, right? Perhaps among how many participants here? More than 100, maybe? some of you may be having problem to consume the dairy products, maybe with lactose intolerance and so on. But I believe everyone is familiar with this. Also with this, meat products, sausage, meatball. I don't sell meatball here, which is very popular and famous in Indonesia and also neighboring countries. Right? I'm sure also all the participants are aware of all these products, and perhaps most of you are consuming this one in daily life. And then also this one, egg-based products from chicken, from ducks, and so on. So basically, all these are the main products from animals, which are known to be very, very rich with proteins. And in fact, that animal products are considered as the main source of, or excellent source for supplying protein intake for the people. And the study on the animal based food is actually to provide the good quality of protein products, which is statistically is relevant to the development of the Human Development Index, HDI, whereby the high consumption rate of the protein is usually accompanied by the high index of the HDI, or Human Development Index. Yeah? Well, of course, it's debatable, but the developed country are always indicated by the high protein intake. But yeah, what I would like to say here is in general, we always put the animal product as in the form of food, but in fact, actually, the functions or the benefit of the animal products is quite wide beyond their nutrition role. It's not only to supply the protein intakes, it's not only to supply the nutrition needs of the human being, but also can benefit more than the nutrition role. So we have to think beyond the nutrition role. So this is what I would like to talk. One of the functions beyond the nutrition roles is the antiviral activity. So before we'll go for detail what kind of proteins that we can find in the animal products that having antiviral activity, let me brief you a bit about the virus itself. Perhaps because some of you are not coming from the relevant background, so I think this is also important to briefly recall back our memories about the virus. What is virus actually? I would say that this is not living things. Yeah? This is not single cell. This is not an organism. Rather, we can call it as a particle actually. Yes, it is particle. That's why you can call it as living things or in non-living. So it's actually in transitions between two things, two properties as a living organisms and non-living organisms. And the size also is in microscopic size, in micrometer scale, whereby it usually virus is higher size than cell organisms like ribosomes and so on. It's higher, but of course it's less than the cell sizes. That's why they can easily infiltrate the cells. And then it's contagious, means that infections, it can create the infections, it can move from one person to another person, or one host cell to another host cells, from one healthy cell to another healthy cells. And that's the things that makes this viruses dangerous. And then what can be the host cell can be any organisms, can be started from the prokaryote organisms, let's say bacteria, and also to animals and humans. So no wonder, don't be surprised that the virus also attacking the bacteria, not only us, and also not only animals in the farm. So if you look into the comparisons of sizings here for the virus, you can see that from the right side is the small size for the atoms, molecules, and then elements and so on. So you can see that the virus is in between the cell organelles and bacteria. Virus ribosomes here, and then bacteria is the pox virus. As an example is in the between bacteria and the cell organelles. And then to observe, usually light microscope is not powerful enough to see the virus. You need a more powerful a microscope, which is usually we can use electron microscope, right? So many studies about the morphology and then shape of the virus were done under electron microscope. All right. So, and then if you look at into the structure, which is important to know because the structure will modulate how the virus infect the host cells, we can see basically the structure is consisted only two major parts: nucleoside and viral envelope. In the nucleosides, you will find the genetic material, as you can find in any organisms, but the virus can contain two types of genetic materials, either DNA or RNA. I believe you already know what is the difference between DNA and RNA. And then this genetic material is protected by a structure called as capsid. And then out of the capsids, we have what's so-called as envelope protein or viral envelope, right? Whereby in this envelope, there are several proteins that involve to protect actually the material genetic of the virus. We have envelope protein, or call it as protein E, membrane protein with spike protein or protein S, and so on. So I believe that during this pandemic COVID, you may heard about S protein, mutation S protein that leads to the different type of variants of COVID and so on, so on. But not all virus having envelope components. Yeah? There are non-envelope virus and envelope virus. It depends. This is the two types of virus depending on the presence of the envelope. So we can see that some virus having envelope, another virus is naked without envelope. So of course, by knowing the structure, the strategy to kill the virus will be different, because the virus with envelope, of course, theoretically will be more stable compared to the virus without envelope. Yeah? And then, of course, the treatment will be also different. And then also the material genetics, as I mentioned earlier, are also different. Some viruses are DNA basis, which means that their genetic material are in DNA form, double helix and so on, all right? And then another group are in the form of RNA virus, which is they have only single-strand RNA. Most of virus in single-strand RNA is quite dangerous or prone to mutations, and because the RNA is prone to mutations, prone to have more mutations compared to the DNA. That's why we can have more variants like influenza virus or COVID that we now still fighting with. So those things are different types of the virus depending on the type of material genetics. All right, and then the capsid structure also different. There are some virus with cubical septs of the capsid, helical sept of the capsid, and complex septs of the capsids. But most of the virus that attacking animal are mostly in cubical septs. Yeah, cubical sept is like hexagonal like that, icosahedral, and so on. So this sept will also benefit. Knowing the septs of the capsid of the virus will also benefit us in order to find the best way to kill the virus by destroying the capsid structure. You remember that the capsid is protecting the material genetic, whereby if you're able to destroy the capsid, means that you will you will release out the material genetic, and then it's easily to be destroyed. So all those things are very important in order to find out the way to combat those viruses. All right, and as I mentioned earlier, that the one that can be the host cell for the virus is not only eukaryotic cell, it's not only animal, it's not only plants, it's not only human, but also single cell of bacteria can also be attacked by or infected by the virus. You can see this figure showing these two types of virus. The left one here, animal virus structure, is the virus that often infecting the eukaryotic cells, eukaryotic host cells. And then the one in the right side here is the virus attacking the bacteria. Call it as bacteriophage. So whenever you heard about bacteriophage, it's referred to the virus that attack to the cell, bacteria cell. So look at the structure; it's quite similar. Only the bacteriophage is quite unique because it has additional structure of neck here, and then tail, sheath, and also tail fiber, which involve in the penetrations of the virus on the cell surface of the bacteria before injecting the material genetic that are located inside of the capsids of this virus. All right. Okay. So we are not worrying about the bacteriophage because the bacteria is not really, that's problem with bacteria, but actually indirectly it will have a problem for us because virus can create the mutations on the bacteria, and then the good bacteria can become bad bacteria due to the mutations due to mutation caused by this virus infections. What we worried is about, what we will have to more worry is about the virus that infecting the eukaryotic cells, including animal. So those viruses that are causing problem for us is mostly the virus that create the disease, call it as viral diseases. And the one that the most common viral diseases among us is common cold or influenza. But actually, the one that you got every day, let's say, there are two things. Cold and influenza is two things different. Cold maybe become allergy, and then influenza is infected, is triggered by the virus infections. So there are several types of the influenza virus that maybe mutate along the time, and then that's why in several developed countries they will always have regular vaccination for the influenza virus. Other than influenza virus, there are some other viral diseases that you can see in the list in this slide: chickenpox, herpes, HIV, and so on, and then including these diseases that currently impacting Indonesia. Call it as mouth and foot disease, or MFD. And also, of course, apart from that, we have coronavirus disease, right? All right, so I would like to briefly mention only two types of virus that are currently popular among us. One is the influenza virus, as one of the most common virus in infecting human being. All right. Influenza virus is actually not only infecting human, but also all the in wide range of the eukaryotic organisms, particularly on animals. We have different types of influenza viruses, A, D, and C, B, which infect different type of organisms. So you can see that in middle here, there are several types of virus that can infect same organisms, can infect different type of organisms. One type of virus that can infect same organism. That's why the zoonotic influenza virus is become serious issue here, including SARS-CoV-2. Right? Well, it's still debatable whether the SARS-CoV-2 is actually coming from the animal or not, but in fact, that there are so many proof that it's coming from the animals, and then we can consider it as a zoonotic influenza virus. All right. Another one is, as I mentioned earlier, that quite serious issue in Indonesia starting from this year is foot and mouth disease, which is actually disease of viruses with capsid icosahedral RNA base. So it's easy to mutate. That's why there are several variants of foot and mouth disease. We got problem with this in Indonesia, I believe also in some other countries maybe. And then this is how actually the animal that being infected by the virus, and then this is how the virus looks like. I don't want to talk more about this, but you can see here that the capsid is quite clear with the spike proteins coming out from the membrane of this this structure, which is actually clear enough to find out the way how to block this virus. But the problem is it's quite contagious with the infection rate is quite high. All right, and then also the problem also this is messenger RNA based that is prone to mutations. That's why recombination sometimes occurs. Recombination is possibly these two different type of virus merge together. They merge their material genetic. That's why we may have FMD virus different types due to the merging of the material genetics during the infections in one host cells. All right, so and we have been living with the virus for many decades. Actually, it's many centuries ago. All right, the virus is not new things for us, but the virus, the problem is always evolved. We can see that, let's say, since smallpox issue, Spanish flu, and so on, even before that, before modern era, we have also problem with other virus. This is just a historical timeline on how the virus creating issue during the human history, and then of course along the development or along the development of the science and technology, we can actually handle the pandemic or handle the virus disease way better. Like what we have now is way better compared to, I mean, where the current pandemic, the people treating the current pandemic is way better compared to the way people treat the Spanish flu in 1918 or 1919. So it's because of science developed more advanced at this time compared to decades ago. So, or centuries ago. So anyway, this is nothing new actually, just virus is keeping developed. All right, so and then that's why we have actually extensive works on people. I mean, researchers have extensive works on how virus infect host cells and so on. And then the theory of the infections or virus replication inside of host cells are not. I mean, it's clear already. Okay, just maybe the detail is different for each of virus. This is the basic route of the virus replications when they infect the host cell, starting from what so-called as attachment phase. This is the virus, all right, with the material genetic inside, and then at first step, it will try to attach to the cell surface of the host cells, and after that, followed by the penetrations step in which the material genetic will be released from the virus particle getting into the cytoplasms, and then this material genetic will be further transcripted and then translated into its protein products, whereby the protein is polypeptides of the product will be necessary or will be important for creating the new particle of the virus, and then all this new particle will be assembled, and then you will get new virus particle inside of the virus. And then after that, it will be released out as a baby of the new virus particle that are be ready for infecting the other type of new cells. All right. Of course, some other defense, our natural defense will work out throughout this process, but it depends on our immunity. But anyway, I won't talk more about this part, but only I would like to show you how it infects the host cell. So the detail, all the virus will infect through the same roads, but there will be different in the detail of each step. Like, okay, virus A may be attachment will using different receptor, virus B using receptor X, and so on, so on. So this is how the video that showing how the penetrations of the virus and then getting into the site of cells, material genetics is being released, right? So this is the animal virus, and on the right side here you can see the bacteria of veg infecting the cell, bacteria cell by injecting the material genetic through the tail sheets here, right? So look at here, the fiber of the tail is used to anchor into the cell surface before they injecting the material genetic. All right. So I said that the route of the replications or infections are all the same for all viruses, just the detail are different. This is an example for HIV virus or influenza virus. You can see that they are different only into the attachment process, whereby the HIV will utilize a receptor called a CD4 protein, all right, as an anchoring agent on the cell surface. So CD4 is the hand that will catch the HIV virus, and then it will allow the virus to get into side cells. Moreover, for the influenza virus, it's not CD4, but sialic acid on the surface of the cells. That's why the susceptibility of animal cell will be different towards different type of the different type of the virus. Like maybe you ask why human is more susceptible against COVID virus compared to the animals, because we have the receptor that able to anchor to this virus, but animal they don't have, or the receptor are quite different with us. That's why they are quite they are more resistant compared to us. Okay, and then why upper respiratory tract is more susceptible for the influenza virus? Oh, because upper respiratory tract having more sialic acid compared to the other type of the cell. So the difference is only on those details. This is SARS-CoV-2. SARS-CoV-2 will bind specifically into what's so-called as ACE2 receptor located on the respiratory tract. Okay, this ACE2 receptor is not found in other type. I mean, minorly found in other type of the cell. That's why SARS-CoV-2 virus will not be able infect the other type of cells apart from the respiratory cells. Then FMDV, FMD virus, it will attach to the cell through what's so-called as integrin receptor. Right. After that, if you have the integrin receptor, if that animal having good integrin receptor, highly expressed integrin receptor that can bind properly to the FMD virus, then that animal will be definitely susceptible towards this type of virus. All right, so the difference are very minor. The flow are quite similar for one virus to the other virus. So by knowing this, we can understand, or we can devise a strategy to block this virus replications. How? Of course, by blocking each of step. You can block either at attachment step, penetration step, replication step, and so on. So that's how actually people working on the development of the antiviral agents is trying to inhibit the virus. If you can block the attachment process, then the virus will not be able to get inside the virus. Or let's say you cannot block the virus attachment. Let's the virus enter the cells, but block the replication so that virus won't be able to produce the polypeptide and so on. So any steps, any essential step you can choose. It's not necessarily to block all the steps. Just find one essential step or critical step along the replications process, right? So this blocking is the crucial, and we can block by using other molecules that can bind to that virus so that they misrecognize the receptor, they misrecognize the other components, other cell components for the replications and so on, right? So this is an example how we block the process. This is FMD virus. FMD virus, we know it infects the cells in bovine, in cattle, by binding to the integrin. This is integrin. We have alpha and beta integrin. This is the FMD virus. Yeah? This is more detailed. People study about the interactions between the integrin and FMD virus. This is more detailed how the amino acids interact among the receptor and the human virus. Okay, if you can block these two, then the FMD virus will not be able to infect. Yes, the virus is still inside the cell, but it's still outside the cell. But if they cannot enter the cell, it's useless. The virus will die because they have no machinery to replicate. They need to be inside of the cells to replicate, right? So by knowing this, then this is a figure where they found a compound of glycan that able to block the interactions between the integrin and the FMD virus. Same thing with COV2, all right? We know that people know that spike protein from the envelope of the SARS-CoV-2 is required to interact to ACE2 receptor here. So if you can block these interactions with any compounds, then you can prevent the infections of the cells by SARS-CoV-2 virus. That's how actually, just an example of how we can inhibit the steps of the replications virus. All right, okay, so do we know what is a virus? We know how they replicate, yeah? And then we know what the steps that we can block it in order to avoid the replications and infections, of course. All right, so now let's see what kind of proteins that we can find in animal-based food. Okay, the animal-based foods here is limited to two major foods that I have mentioned earlier. We have milk, we have eggs, and meat. All right, so milk is actually not protein source. It is actually for majorly known for sugar, because it contains a lot of lactose. Lactose is this disaccharide, which is actually good source for energy for us, because it can be break down into the two monosaccharide and then absorb and then become good ATP sources. But apart from the lactose, milk also known to contain several major proteins, including casein, right? Okay, casein. This is the major proteins from milk. And in fact, if you see, the protein inside of milk is not much. Actually, it's only 3%. Okay, less than fat and less than lactose mineral. That's why it's actually not good source, but it's still there. The protein is still there. All right, and this is the complete profile of the protein that you can find in milk. We have major protein casein. We have several type of casein, alpha, beta, gamma, and so on, right? And then non-casein protein, we call it usually as whey proteins. Okay, and inside of whey proteins, we have alpha, beta, and so on. So I believe Pa Arifin knows better about this. Can explain more about this later on. Right. So basically, there are a lot of type of proteins, casein, lactalbumin, lactoglobulin, even lactoferrin, and so on. So on. You imagine if you put milk on the table for maybe one day or a couple hours, maybe, sometimes you can see the separations of the milk, all right, because of the maybe the gradations of the protein that enable to be emulsifier on the milk. Then you can the oil and then the protein part will be separated. Then you can see whey proteins there, okay, that are already separated with the casein. So, or you can, let's say, you can add the acid, and then the casein will be coagulated. The other protein is still in liquid form. That liquid form, we call it as also as whey proteins. And inside of the liquid form, we can find other type of proteins, non-casein proteins like lactalbumin, beta-lactoglobulin, and so on, so on. Okay. All right. This is proteins from beef, or meat. Meat is very well known to be the source of proteins. Okay. If you compare the content of the organic molecules inside of the meat, the major organic molecules will be the protein for any type of protein, beef, lamb, pork, veal, pen, and so on. It doesn't matter. All right. Basically, the protein inside of the meat can be classified into two groups. One is myofibril proteins. You look at into the structure of this protein in the right side here. Myofibril is you can see that the muscle is actually composed by hundred thousands of muscle fiber here. So inside of muscle fiber, there are myofilament, all right? Those who study about meat science, perhaps you understand about this, right? That myofilament is involved in the integrity of the muscle structure and also contraction. You can find myosins, actin, and so on. So the protein that's located in the myofilaments is called as myofibril protein. Usually, it's not well dissolved in water. And then second group is called a stromal protein. Stromal protein is the one that located in this area, in the connective tissue. Actually, the one that connecting the muscles to the bone is one type of the connecting tissue, but not only that one. Actually, each of muscle cell is also wrapped by the connective tissue. We have endomysium, epimysium, perimysiums. All are actually stromal proteins. And then the sarcoplasmic reticulum is the one that located in cytoplasms of the muscle cell. But in muscle cell, we don't call it a cytoplasms. We call it the sarcoplasms. It's just naming, different name. So all the other protein that are mostly that located in the sarcoplasms, we call it the sarcoplasmic reticulum proteins, and indicated by their high solubility in water. Okay, so all those proteins are good for protein intakes, of course. The another one is egg proteins. Or if you open an egg, crack an egg, you will see the yolk here, an egg white part or albumin. Protein will deposit mostly in white proteins. You can see that in the yolk is very, very low contents of the protein. Most of the protein deposit in the egg white. Right, and in egg white is the proteins inside of in the egg is quite unique because it's very good quality. The biological value is close to 100. If you look at in this table, 100 means that all the proteins inside in the egg can be absorbed by our digestive tract. That's why, as a standard of the protein quality, egg is often used, and then it's way better compared to the other protein sources, including non-animal protein sources. All right, this is the major protein that you can find in egg white parts. You can see ovalbumin, conalbumin, ofomucin, and so on. Actually, based on the proteomic studies, there are more than 100 proteins located in the egg white parts, but the major one is only less than 10 proteins. Yeah, major one is the one that, in terms of quantity, are remarkably high. Okay, all right. So this is also the list of the major protein with the of different functions. Yeah, basically they have different functions because egg is biologically is created to protect the embryo of the chicken, of the birds. That's why some proteins will serve as the source of energy. Some proteins serve as the protections, antibacteria, and so on, so on. So they have different functions. All right, so let's see what type of proteins that been reported to have antiviral activity. The first one is lactoferrin or lactotransferrin. All right, lactoferrin, whenever you see lacto here, is actually referring to milk, but it's not always. It's not necessarily, but mostly. All right, so this is mainly this type of protein mainly found in dairy products. You can go for milk and or other fermentations product, and it has moderate size, about 80 kilodalton, and then the content, the concentrations of lactoferrin inside of colostrum, the early milk secreted by the cattle, the cow, will be higher, compared to the at lactation period. All right, this protein is characterized by ability to bind to iron. Yeah, that's why they call it a ferrin. Ferrin here is referring to Fe. You can see that the amino acids here is quite nice, because of the presence of tyrosines and then other hydrophilic residue that able to coordinate the Fa metal ions. All right, and then the ability to bind to Fe here will help this protein to regulate how the ion will be absorbed by the intestine and also get into the body. That's why they can control our needs to the Fe element or iron elements. And also it can kill bacteria. Why? Because the Fe or iron from the bacteria can be killed, can be absorbed by this bacteria, by this protein, and then the bacteria will be deficient in Fe elements and iron elements, and they will die because iron is essential for the bacteria cells. All right, when the a researcher tried the lactoferrins against the herpes simplex virus. This is the virus electromicrograph, and this is the symptoms that you can observe on the skins. Hopefully, none of you will suffer from this disease someday. All right, you can see that lactoferrin can dramatically, significantly reduce the number of virus in titers. 1% and is in concentrations dependent manner. The more lactoferrin you add it into the infected virus, the more virus being killed. And then also the infections, the killing time of the virus by lactoferrin is stable until about 1 week after the introductions or administrations of the lactoferrin into the cells. All right, and then, but interestingly, different source of transferrin are having different inhibitions activity. This is from overtransferrin, this is human lactoferrin, and this is bovine lactoferrin. Look at this, that bovine lactoferrin has strongest activity against the herpes virus. It can block the virus replications way higher compared to human lactoferrin, and there's overtransferrin. Overtransferrin is homologs of lactoferrin can found in egg white. So does it mean overtransferrin is bad? No, it's not bad, but just because of maybe it's good for other type of virus, right? So you can see that a bovine lactovirus is way better in inhibiting the virus compared to human lactoferrin. All right, how the lactoferrin block the virus actually? This is the herpes simplex virus. They will infect the host cell through what's so-called as GAG binding site located on envelope protein, this one. All right, so this one will interact with the sialic acids or sugar parts of the host cells. So the lactoferrin will protect this sugar part of the host cell. That's why it will not be accessible by GAG binding sites on envelope proteins, and that's why the virus will not be able to attach, and then the infections will be avoided, right? And then another activity is again HIV virus. All right, you can see here that lactoferrin from bovine milk, of course, it reduced the HIV replications in concentrations dependent manner. You can see in the A panel and B panel here, right? And then, but it's still concentrations wise is still high, but I think it's just a matter of the optimizations later on. All right. Same thing go for rotavirus. Rotavirus is the one that mostly attacking the kids and then creating the diarrhea. Okay, this is the things when we have to introduce the lactoferrin. This is the control without any lactoferrin. This is lactoferrin added before attachment of the virus. This is lactoferrin, the C, during the attachment of the virus. This is lactoferrin added after the attachment. You can see that the best way we treat the cells is after the after the attachment of the virus itself. What does it mean? This means that lactoferrin cannot be used as preventive, but lactoferrin can be used as a drug to cure the cells, the virus attachment to the host cells. Yeah, at least for the rotavirus. All right, and then hepatitis C virus. This is interesting. Okay, what they did is they treated the cells with the hepatitis C virus. All right, with infected. Okay, and then they added with the different lactoferrin from different organisms. Yeah, human, camel, bovine, and sheep. You can see that if the virus successfully infect the cells, you will see this band. This band indicate the presence of material genetic of that hepatitis C virus. You can see in A, okay, in the presence of 0.25 milligram per ml of lactoferrin, the virus, hepatitis C virus is still happily alive. 0.5 milligram per ml, camel can block the virus. Camel lactoferrin can block the virus. At 1 milligram per ml, all lactoferrin from all organisms can completely block the virus by indicating by the absence of the material genetic of the virus itself. So means that every lactoferrin will have their own specificity, their own activity, different activity against the virus. All right. The next is, I cannot move. All right. Okay. So how do lactoferrin works in inhibiting the virus? Different virus will be inhibited with different mechanism. For example, for HIV, as I mentioned earlier, actually, it blocks the attachments of the HIV virus to the host cells. In particular, the attachment is mediated by the GP120 proteins located at the cells, located at the envelope of the HIV virus. The GP120 will be caught by lactoferrin. So GP120 will be like the eyes that be blind because of cover by the lactoferrin. For the rotavirus, it's different. The lactoferrin will possibly prevent the attachment of rotavirus to the cell epithelia. You know that when the virus are unable to access cell epithelia, then bad things will happen, right? So then the lactovirus will occupy the cell epithelia, and then the virus will not be able to access. And then at the end also, the T cell is the cell immunity, or we call it the cell lymphocyte cell, the one that having memory to attack the next infections of the virus, will be protected by the lactoferrin by avoiding the attachment of the virus against the T cells. Same thing with this. If this one is lactoferrin in the epithelial cells, so the virus will not be able to access, and hepatocyte cell also will be protected by the lactoferrin from the disruptions of the virus. All right, and then lactoferrin to be specific for hepatitis C virus by inhibitions by lactoferrin. This is also done. The inhibition also done by preventions the attachment. In particular, researcher found that E2 and E1 envelope glycoprotein of this HIV virus is being blocked, yeah, by the lactoferrin. So lactoferrin will bind to this E1 and E2 envelope glycoprotein, and that's why this glycoprotein will be blind. They don't know where is the receptor, and they won't be able to attach to the host cells. All right. This is other virus that being blocked by lactoferrin experimentally. Yeah? Respiratory common cold, and then influenza virus, adenovirus, viral gastroenteritis, yeah? And so on. So, right. So, and then I think you can find this type of products for milk powder that being enriched by the lactoferrin, eh? And because the functional properties of the lactoferrin. So, all right. Apart from the lactoferrin, actually, we can produce what so-called as bioactive peptides from the milk, whereby actually bioactive peptides we can produce by hydrolyzing the major proteins. Look at this, yeah. milk protein. Of course, we have casein and whey proteins. We can digest all these proteins by either process fermentation, so hydrolysis, or by using proteases. And then, of course, all this protein will be fragmented into short fragments of polypeptides. And usually, if you are lucky, some peptides produced from these digestions will exhibit different activity that extremely different to the parent's protein here. Some peptides will have antibacterial activity, some other peptides having antioxidants or antiviral activity. So basically, this paper is very interesting, whereby it review the fragmentations of the milk proteins by using proteases, and they confound some peptides that can have anti-HIV activity here. Right? The negative means that it has no activity. The positive one having strong activity, positive minus like this having moderate activity against the HIV virus. So that depending on the peptides it produce and the properties of the peptide, then the antiviral activity will also be different. The point is, yeah, you can produce the bioactive peptide with antiviral activity. All right, lysozymes is from egg white proteins. Right, lysozyme is basically one of major proteins in egg white proteins, and is well known for its antibacterial activity. How the lysozyme works is basically by degrading the peptidoglycan. I believe you all know that peptidoglycans exist in either gram-positive or gram-negative, just location are different. In gram-negative bacteria, peptidoglycan is being protected by outer membrane, and it's very thin. Okay, protected by outer cell membrane. Meanwhile, for gram-positive, it's thick peptidoglycan, thicker peptidoglycan compared to gram-negative, and then it's naked. There is no outer cell membrane protecting this peptidoglycan. The lysozymes will cleave the bond between N-acetylmuramic acid and N-acetyl glucosamine of the peptidoglycan. When it's disrupted, of course, the peptidoglycan structure will be gone, and then there are so many pores in the surface cell of the bacteria, and then bacteria will die. Okay, but apart from its antibacterial activity, lysozyme is also been reported to have what's called as antiviral activity. Let's see what kind of antiviral activity being reported for the lysozyme. This one is a diarrhea virus attacking mostly on cattle, bovine. All right, you can see that lysozymes compared to nisins, CC and EC is quite similar. It is ability to kill the virus up to 50% of the viability of the virus. You can say lysozyme can kill the this virus better than nisins, but similar to lactoferrin. And so it works well for the diarrhea virus. And then, but the problem is people still arguing how the lysozymes actually kill the virus. But one theory is due to the ability was proposed in which the lysozyme has ability to degrade the material genetic of the virus. So when the virus is, so in this case, the lysozyme will let the virus infect and get into the cell. But once the virus release the material genetic, the lysozyme will chop down the material genetic so that the virus will not be able to produce the polypeptide from its material genetic, and there is no more virus particle newly produced. And then another one is against HIV. You can see that lysozyme from different type of eggs, quail, melligrass, and hen can block the HIV virus and concentration dependent manner quite nice here using CD4 receptor. And then it's interesting because the lysozyme here is not blocking, it's not degrading the RNA or the RNA of the virus, but it's blocked the GP41 proteins, which is important for attachment to the CD4 receptor. So you see, one protein will use different mechanisms to block different type of the virus. Okay, so this is how this lysozyme block the HIV virus. This is GP41 protein. Sorry, GP41 protein is required to attach to the host cell through CD4 receptor. This is CD4 receptor. Okay, so the lysozyme basically will interfere this area so that the interactions between the virus and CD4 receptor will be prevented. All right, this is lysozyme, again, poliovirus. You can see this is poliovirus without lysozymes. and then this is cell with lysozyme and poliovirus. This is cell without lysozyme and infected by poliovirus. You can see that the cell is die, while you can, in the left side here, the healthy cell is obviously observed under the microscope, indicating that the lysozyme is able to protect the cells against the poliovirus. And in fact, because of antibacterial activity and antivirus activity of lysozymes, some like portable air purifier produced in China is equipped with the lysozymes on the filter, so that the lysozyme can kill the bacteria that being filtered by this air purifier. Okay, I don't think it's available in Indonesia now. All right, so and then other egg white protein that's been reported to have antiviral activities, including ovomucin, ovotransferrin, ovalbumin, ovomucin, and ovo inhibitor. Rotavirus, NDV is Newcastle disease virus. I think those working with poultry know well about this virus. Yeah? In Indo, we call it Patelo, is it? And IV is influenza virus. You can see that the inhibitions activity of each protein are varied. Okay, some are good for rotavirus, some are bad for, some are good only for a Newcastle disease virus, and some are good for influenza virus. So each of protein will have their own specificity. Okay, right. All right, and then egg white proteins for ovomucin and ovotransferrin and ovalbumin. Interestingly, they belongs to what's called as glycoproteins. Glycoprotein is the proteins that are able to conjugate, to bind to the sugar group or carbohydrate group. And we know that the spike protein, the cell surface or envelope proteins of the virus is always glycosylated or bind to the sugar. You imagine that if this virus is able to interfere the sugar moieties, sugar parts of the virus, then that virus will be blind, because it's like your eyes is being covered by something that covering your sight. Then you can do your activity normally. So that's how actually these glycoproteins can prevent the infections of the virus, basically by blocking all the carbohydrate parts on the surface of the virus. Yeah. Okay. All right, and then interferon is very interesting. Interferon you can find in all the animals, including, but most of the interferon can be easily found in bovine. This is the product of the interferon supplement. You go to drugstore, you can find some local interferon supplements. Immunity, because what is interferon actually? Interferon is actually proteins that important to give a signal to kill the virus that getting into the cell. So basically, all the cells, all the organism will produce interferon, just only different intensity, different activity. So if you imagine, if we can purify the interferon, collect the interferon from the vine, let's say, and then apply to the sick cell, to any cells that being infected by virus, there is a possibility that the virus will be then saved by the cell will be cured by the virus replications. Right. This is another one, alpha-lactalbumin, majorly found in milk, structure similar with lysozymes, and then majorly inhibit the herpes virus. How it inhibit herpes virus is very wide mechanisms. It can disrupt the viral envelope, restraining the interactions so that the virus cannot enter the host cells, inhibiting the transcriptional activity, so means that the material genetic cannot be translated into the polypeptides, and then inhibiting hydrolytic activity of herpes virus protease. When the polypeptide is produced by the virus, this polypeptide will be assembled to form a new particle of virus. It requires protease. So this protease is like cutter. So if this cutter is being blocked, then the new particle will also not be formed. All right, this is other things. Beta-lactoglobulin, again, herpes virus, and then HIV virus. Okay, I don't go for detail because of timing. And influenza virus, also being blocked by beta-lactoglobulin, and then also the influenza virus of H1N1. Right. So what is the opportunity for knowing all this antiviral activity of this animal-based protein? The first thing is, okay, let's see. This is published last year. It's very relevant to what happened in Indonesia this time about the outbreak of the foot and mouth disease. You can see that lysozymes is interestingly able to partially inactivate. Partially inactivate is not completely inactivate, but have partial antiviral activity against foot and mouth disease strains, different strain. We have here A strain, B strain, and Taiwan strain, Asia strain, and so on. There are so many strains here. So when lysozymes is being heated up, it can block partially the virus replications, right? Yeah, well, up to now, it's not being able to completely block the virus, but maybe by optimizations of the treatment, maybe we can block the FMD virus completely. So egg white can be used for FMD virus. It's just question mark. I leave it for you to answer. All right, same thing with the current pandemic of COVID-19. Right, as I mentioned, that some animal proteins are glycosylated. Right, is glycosylated is usually is able to. I mean, the glycosylated proteins were able to block the protein E of the virus. Right, this is the protein E. You imagine if we can block this protein E, then the envelope of virus will be disrupted and so on. So, does it mean that we can use some proteins like ovomucate, ovotransferrin, and so on to block the SARS-CoV-2 E proteins? That also a questions I left open for you to all to answer. Something, this is spike protein. Spike protein is known to be very glycosylated. This is glycoproteins. So, and then we know earlier that some proteins from egg white also very sensitive, also glycosylated, and also can bind to the glucose or carbohydrate part of the spike protein of other virus. So maybe we can use that protein for blocking the S protein of SARS-CoV-2. Maybe, but still I leave it as questions mark. So in fact that some protein-based drugs are being developed for the treatment of the current COVID-19 diseases. Yeah? Lopinavir, ritonavir, they all are working differently, inhibiting different type of virus replications and so on. Yeah? Interferon, remdesivir, hydrochloroquine, and so on. Yeah? So we can block any steps with any proteins that having the most sensitive blocking activity. As a conclusions, we know that animal-based protein having promising antiviral activities against some virus with different mechanisms. Same proteins may have different mechanisms to block the virus. So in order to further develop those proteins, if we want to further develop those proteins as a drug, we need two things here. One thing is we have to have solid fundamental research on those proteins. Second thing is we have to think beyond the nutrition. We have to stop thinking that it's only about food, but animal products can be used for more than that one, right? By this, I would like to close my talk. Thank you for the time, Pak Arifin. Thank you very much. I'm open for the questions and comments, if any.
Yeah, thank you so much, Pak Cahyo. It's very interesting topic. I think we have two questions in our chat room, but I offer the participant who ask directly to Pak Cahyo, please. You can use raise hand first. Okay. Or we can read the question at the chat room, Pak Cahyo. For the first one, maybe from Johannes Bayu. Do you want ask directly to Pak Cahyo, maybe? Are you there? It's okay. I will read it for you, Pak Cahyo.
Okay. I can read.
Okay.
All right. Do I need to read it loudly?
Yep.
Okay, so the first question is thank you, Bayu, right? So currently FMD livestock is non-natural disaster in Indonesia, but the number of vaccine is still not evenly distributed in all regions, especially in areas that are still free of FMD. Yeah, is there a way to develop vaccine for existing vaccine to speed up based on the yes, definitely. Basically, we are actually technologically for vaccine development is the country Indonesia should be able to develop that one. Just I think we need political support from the government to be able to develop that one. And then also we cannot rely on the vaccine that we got it from outside, because maybe, as I mentioned, that FMD virus is prone to recombinant, prone to mutate because of the recombinations. Maybe specific vaccine that can specifically recognize the part of the virus is very important. And then the one that may be infecting our cattle in our country may be quite different with the one that available in other countries. So that's why I think, yeah, let's say if we know that, okay, the theory is like this. We know that FMD virus is affecting by, infecting through the interactions between its virus part to the integrin. So it means that we can find the fragment of integrin that can, we can synthesize the polypeptide mimicking integrin, so that can bind to the part of the FMD virus. So it's like one persons want to meet B persons, okay, but we ask someone else, C person, to mimic, to pretend like B person, so that A will meet C, but he or she doesn't know that he actually meeting C. He or she thinks that he is meeting with B person. So that's how actually we can create the vaccines, or drug. So we can just find, I think, for me, just easiest way is just find out what is the specific region in the integrin, and then that's created as either as epitope for the vaccines or for the drug. Yes, possible. Is there any way cattle can survive from FMD without vaccines? Biosecurity. The only way to, okay, I'm not the I'm not veterinary science, but what it's common sense that virus is difficult to be treated. Among microorganisms, virus is the most difficult to be treated. Bacteria, we have antibiotic and so on. Antivirus is also available, but not virus is always mutated. So the only way is we optimize biosecurity, and then also biosecurity here is to prevent, because virus is contagious. Virus can only replicate when they found the healthy cells. So when we implement the good biosecurity, means that we reduce the chance for virus to find the healthy cells to replicate. That's it. And then, of course, apart from vaccines is drug, antiviral drug. Vaccine and drug are two different things. Vaccine is to prevent, drug is to cure. After the one that I talked just now is the protein is for the drug, not for the vaccine itself. But the theory are the same. Basically, we are trying to protecting the cells itself. I hope it answers, Pak Budi, Pak Bayu. And then, can we go to the next questions?
Yes, please. From
Rafida. Next is from Rafida.
Yep.
Can we find animal-based protein in plants? So, some animal-based protein having homologs in plants, but I think the similarity will be different. There are so many proteins, but the one that I mentioned just now, like the major proteins in milk, caseins, like lactoferrin or lysozyme of a mucoid is very unique. You can find in plants, but others, maybe the minor proteins, you can find. But I don't think the similarity will be 100%. Some proteins are quite unique to be only found in animals. Some other can be found throughout the higher level organisms, including plants, but different, I think, at some degree similarity. I hope it answers. And then Gasani, I want to ask a question. So based on your lecture earlier, milk is actually more to lactose source rather than protein source. Then why does most of the milk ads say the milk is good source of protein? Also, there is lactose-free milk for the lactose intolerant people. So when they drink it, it will gain what will they gain because there are no lactose in it? Okay, or will it be better for them to drink plant? All right, okay. Based on the concentration-wise, lactose carbohydrate is higher compared to the protein in milk, but protein is there. So basically, when you drink milk, you still can get proteins. Okay, but you compare with other animal products like meat or egg. Of course, meat and egg are having higher protein level compared to the milk. Okay, meat and eggs. Yeah, all right. So in regard to the second concerns on lactose free, yeah, when the basically the lactose intolerance people having problem when they drink the milk, the problem of drinking the milk is due to the presence of lactose. The lactose has to be degraded into glucose and galactoside, all right, in order to be able to be absorbed by our small intestine. And it can be done by the enzymes called as lactase. All right, the problem is not everyone having enough level of lactase. Lactase productions will decrease along the age. All right, when you get older, the lactase will get reduced. That's why at certain age you will get lactose intolerance. Okay, or some people genetically having problem with that lactase problem. All right, so when the product remove lactose from the milk, means that people will be safe drinking that milk because the lactose is not exist anymore. So what is benefit for those kind of people? They still can get casein, they still can get the essential fatty acid and so on. So other nutrient compounds apart from lactose is still in the milk, so they still can get the benefit of the milk. Actually, the best way for the lactose intolerance people to get the benefit from the milk is by consuming fermented milk, because in fermented milk, lactose is already being degraded to the monosaccharide by the fermented by the bacteria. So that will be good, because they will still get the source of the carbohydrate apart from the other apart from the other nutrients compounds. I hope it answer Suraya. And then
for this question, from Haslina, Malaysia.
I'm just wondering if a compound is present in food that can inhibit the virus activity. Will it work better as small molecule in drug, or it can be a good preventive treatment by eating? All right, this is a good question. In order to be able to prevent the virus, that protein has to be in active form, in functional form. If you eat protein, basically what happens is the protein will be degraded in your stomach. You have acids there, very low acid, and then also having a lot of proteases. So basically all the proteins that enter your stomach will be degraded, and then will lose the functions. So eating the antiviral protein is not a good idea unless that protein is being protected by encapsulation and so on. So the protein that I mentioned just now is not for eating way. It's not for oral administration, but maybe by injections or other delivery routes, maybe. But we have to do more research on that one. Yeah. Is that okay?
Yep. From Desti.
Desti, does FMD affect cow's milk production, and what about the human milk? I can't answer this one, but from my reading, the cows that being infected by FMD having low lactations, low productions level on their milk. Yeah, so I think. What about the protein contents? I have no idea about that. Maybe Bu Windy can answer this one later on. All right, sorry, Desti, I may not be able to answer completely. And then from South Korea, can dietary proteins antiviral activity can be affected by digestion? How? Yes, correct. As my answer earlier to Haslina, is it when the proteins in the diet enter the stomach, then the problem is the low acids and the proteases inside of the stomach will completely degrade your proteins. So that's why it's not good idea to consume the protein as a drug by oral. Unless there is a way to protect it by encapsulation whatsoever. Arifin did encapsulations in his thesis work, so maybe he can elaborate more. But anyway, there is a technique for that one. All right. So by consuming in naked form without any protections, it's quite risky. The protein will be degraded inside of your stomach and other parts of the digestive tract. Yeah. So you have to protect it.
Okay, for the last question, I think Pak Cahya from
Pelangi. I want to ask, as we know that there is still no cure for HIV, is there any possibility that antiviral protein that has been mentioned? Yes, could be a drug that can cure HIV. Yes, okay. Theoretically, we can. But the journey to be developed as drug is very, very long. What I mentioned earlier in my slide is at lab level, at plant, in the tubes. I don't think, not all of them go to a clinical trial, animal model, and so on. So that will be a long way to go. Theoretically, I mean, okay, let's say you found the antibacterial compound or antiviral compound, you test it in the lab, all right? You got the good activity, but after that, actually, you have to check it again using the animal model, and then after that, clinical trial using patients, and then after that, you have to check the safety level and so on. So out of usually in the drug research, out of 10,000 candidates or 100,000 candidates, you will get only 1 or 2. 100,000 candidates that having antiviral activity in the lab that come to the drug in the market, maybe only 1 or 2 compounds only. That's quite challenging. But what I want to say here is what we have known in the lab have to be followed up by the next research. That's the thing. Exploration is very important to give us more candidates to further study. I hope it's clear enough.
Yep.
I think
your explanation is very clear, Pak Cahyo. I think enough for this class, maybe. Thank you, Pak Cahyo. Thank you so much for your time. I hope you're still with us for a while to submit a certificate from Prof. Jakaria as a Secretary of Animal Production and Technology Department, Animal Science Faculty, IPB University. Jakaria, you're there?
Okay. Thank you. Thank you, Pak Arifin. Can you hear my voice?
Yes, clearly.
Okay. Okay. On behalf of organizing committee, I would like to give a certificate of appreciation to Dr. Cahyo Budiman for his in teaching a topic regarding animal-based protein for fighting the virus. Thank you, Dr. Cahyo Budiman.
Thank you, Prof. Jack.
Thank you so much, Prof. Jakaria and Pak Cahyo. But before we leave, we can take a picture together. Maybe the committee can provide it.
Okay, thank you, Pak Arifin. We have 4 screen. Please, the screen 1. Okay, we can take a picture. 1, 2, 3. Okay, the second one. 1, 2, 3. Okay, the third screen. 1, 2, 3, and the last one. 1, 2, 3. Okay, thank you, Pak Arifin.
Okay, thank you, Mas Dimas. Thank you so much, Pak Cahyo and Prof. Jakaria and all of the participants. We meet again for next week. So see you soon. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.